Chapter 14, Sensory Systems. This is another one of those epic chapters. This is one of the chapters where I'm going to diverge from the textbook significantly. And that's because while science knows quite a lot about the general sensory systems, of those, the most important is pain. So I'm going to be focusing on that and how your patients may build up tolerance and dependence to pain medications. We'll talk about the multiple types of pain, and there's going to be an anatomical description that accounts for different pain pathways. We'll start off with some generalizations and discuss the difference between sensation versus perception. That's going to be a running theme throughout the next couple of hours. We're going to change the central dogma to the nervous system a little bit. In Chapter 12, I said that neurons did not undergo mitosis. And today I'm going to tell you that, well, sometimes they do. Not often, but it is possible. We'll talk about taste maps. If you've ever learned about the regions of the tongue sensitive to different types of tastes, you've probably been taught something incorrectly. We'll learn why salt is a flavor enhancer. And if you were to leave salt out of foods that don't taste salty, like a cake or some cookies, they would taste gross, even though you don't actually taste the salt when it's in there. And we'll also talk about how spiciness is technically not a flavor. It's not something that our tongue picks up on taste buds. And once again, we'll go back to the difference between sensation versus perception. To do all of that, we'll start with some basic overview of different sensory receptors, talk about how they can adapt to signals, and then we're going to classify them. And we'll begin with the general senses and move on to some of the special senses. We'll talk about the ways that sensory information reaches the brain, and in general, it's fairly consistent. There's going to be a first order, a second order, and a third order neuron. And then we'll move on to the special senses, at least smell and taste in this first part. So our sensory system is the afferent division of our nervous system. That means that neurons pick up information and send it towards the CNS. This is in contrast to the efferent division, which controlled my skeletal muscle and that whole autonomic nervous system that we discussed in the previous lecture. We're going to need to discuss the difference between sensation versus perception. If you look at this image right now, your senses are telling you that this is a five-legged elephant, but you probably perceived it as a regular old elephant. But sure enough, we've got 1, 2, 3, 4, 5 feet there. So that's the difference between sensation and perception. Sensation means the information literally hitting the dendrites of our sensory neurons, whereas perception is how our brain interprets that information. And there's often going to be a difference between these two. Listen to this sound and see whether it matches my description for the sensation or perception. What you are listening to was a shepherd tone. It was actually 3 pitches happening at the same time, but how you perceived it was quite possibly as a single sound that was continually decreasing in pitch. If you go on YouTube, you can find other examples of these that last for hours, and it'll just go on and on and on. But what your brain perceives as a single pitch decreasing in tone is actually 3 pitches that get louder and then quieter, and then a new one starts. This is an auditory example of a difference between sensation and perception. Today we're going to start by discussing the general senses. These are ones that only involve dendrites attached to neurons sending information to the CNS. After about an hour or so, we'll move on to the special senses. These require extra organs to help the dendrites attached to neurons to send information to the CNS. In either case, we've got sensory receptors. That's the name for the cells that detect information. Watch out, we've already used the word receptor to mean the proteins that we find on the dendrites that can bind to chemicals like acetylcholine and norepinephrine. We're now using the same word receptor to not only mean the proteins that we find on these cells, but the entire cell itself. So these sensory cells need to convert information from one form, such as light or sound, into action potentials. And they're always going to code the intensity of that signal, whether it's a bright light or a loud sound, into higher frequency of action potentials, and send this information to the central nervous system. Where it goes in the CNS will determine how it is perceived. Next in our list of general concepts that will be true over the next 2 hours is the difference between receptor sensitivity and receptive field. Receptor sensitivity describes how capable a neuron is of detecting a small signal. On the other hand, the receptive field describes how wide of an area the dendrites extend for that one neuron. In general, a broader receptive field leads to a higher sensitivity. If you have a large receptive field, that means you've got a lot more dendrites. And if you've got more dendrites, that means you've got an increased capability of having small signals summed together to bring the axon hillock to threshold. On the other hand, with a narrower receptive field, you've got fewer chances for summation, but this is going to lead to higher resolution. Story time. Back when I was taking anatomy class, I had a really cute lab partner, and it was our job one day to map out the receptive fields of our touch receptors across our skin. So I had to take a couple of toothpicks and poke my lab partner's skin with them until she was able to determine that it was actually 2 toothpicks. As we see in the upper scenario, for the broad receptive fields, if I were to poke a toothpick here and a toothpick here, those would both be detected by the same neuron. And that neuron either fires action potentials or it doesn't. It can't tell you how many dendrites have been stimulated, just that it's been brought to threshold. That was on our backs and sides and other less sensitive places of our body. It's hard to determine the difference between 1 toothpick and 2 toothpicks. On the other hand, on the fingertips, our dendrites have much narrower receptive fields, and they are capable of determining the difference between 1 toothpick and 2 toothpicks. Stimulating this neuron here, it might fire an action potential, and this one over here might fire an action potential, with one in between not firing an action potential, and then your brain would register that this was 2 individual pokes rather than a single poke. Now, this was a very fun class for me because, as I mentioned, my lab partner was very cute, and she had to lift up various parts of her clothing so that I could poke her with toothpicks on her back, her belly, her thighs, etc. And years later, I gained a significant enough interest in anatomy that I'm now teaching it. But enough about cute lab partners. Let's talk a little bit about resolution. I've got a couple of different pictures of Mario there at a very different resolution. Higher resolution means the pixels are smaller. The benefit of having really large pixels is you have more chances for summation. So this lower neuron here on the left has a wider receptive field. It could have a bunch of dendrites stimulated, and they could all sum together, and this neuron might fire an action potential, telling you that it saw something, but I wasn't really sure what it saw. On the right-hand side, if I have a bunch of neurons with a much narrower receptive field, this will give me higher resolution. It's more pixels, but I don't get any chance for summation here. So if the signal is really dim, I may get no information reaching the brain at all. It would take a much larger signal for my brain to become aware of it, but when it did, it would be a high-resolution image. Next in our general overview is the concept of adaptation. Many neurons, not all, but many neurons are capable of adapting to signals that do not change. Meaning if their dendrites are continuously stimulated, the neuron may start by firing action potentials, but eventually it'll stop firing action potentials, and information will no longer reach the brain. It has adapted to a stimulus that is not changing. Receptors that do that are called phasic receptors. They are going to adapt to signals, and they're only going to send information to the brain when something changes. In contrast to that, tonic receptors do not adapt. They are going to constantly send information to the brain, even if it's not changing. Couple of quick examples. Pain tends to be tonic, meaning if you injure yourself, those pain receptors are going to keep sending information to your brain, and you're going to keep feeling the pain for hours and hours, or days and days, until the repair process has been completed and the injury is no longer there. On the other hand, many of our pressure receptors in our skin are phasic. They're only going to tell us when things change. You were alerted to the weight of your shirt when you put it on in the morning, for instance, but those neurons do not keep telling your brain that you've got a shirt on all day long. They're only going to tell your brain that something has changed when you take that shirt off. There are two major mechanisms by which neurons can adapt to signals that don't change. So if we have a neuron here with a few neurotransmitter receptors, and these are getting a signal that is not changing, this is going to stimulate the neuron. But ultimately, we want to quit responding to this signal, even when it's still there. One way that neurons can do this is by moving some of those receptors into a vesicle and pulling that vesicle into the cell, so that even though the signal is still there on the extracellular surface, most of the receptors have been internalized. We can call this activity-dependent receptor internalization. It means that as receptor proteins are active for long periods of time, they tend to become internalized or moved inside the cell. Our second mechanism is that receptors can often activate enzymes that are capable of phosphorylating those receptors, which changes their shape. And it can change their shape enough that the active site no longer fits the chemical that should bind to it. And even though that chemical, or whatever signal that it is still there, those receptors on the dendrites are no longer responding. We call that activity-dependent receptor inactivation. It's still dependent upon sustained activity. We have to stimulate that receptor protein for a while, but then it tends to become inactivated by a chemical reaction. Second messengers are really good at doing this. Many of our G protein coupled receptors, for instance, after activating adenylate cyclase, which can make cyclic AMP, which can do things like open up ion channels, some of the cyclic AMP may activate a kinase called pKa, which can phosphorylate the receptor, which prevents it from activating more G proteins. So even though the signal is still there, we're not doing anything in response to it. Next, in our general overview of things that are common amongst all of our sensory systems, is the pathway in which information actually reaches the brain. In general, most of our senses follow a 3-neuron pathway to reach the cortex. The first neuron is the actual sensory neuron. It has its dendrites out in our body somewhere, and it converts one type of information, like pressure or light, into action potentials. These action potentials travel up the axon, which will extend to the dorsal roots and connect to the spinal cord and synapse in the dorsal horn. A second-order neuron has its dendrites there, and its axon would travel up the dorsal column to reach the thalamus. The thalamus was our relay center for most sensory information. If the thalamus decides that information is important enough, a third-order neuron, whose dendrites and cell body are in the thalamus, would become activated, but its axon would relay this information to part of the cortex, and we would become consciously aware of this sense, meaning we would perceive it. The thalamus doesn't have to relay this information to the cortex. We are receiving lots of information all of the time, and we aren't paying attention to all of it. There's a lot of sounds and smells and sights right now which you're probably not paying a heck of a lot of attention to. Maybe you've got a heater or an air conditioner in your room right now making a low humming sound, and your brain has been tuning out that information, at least until I started talking about it. Now suddenly your thalamus thinks it might be important, and you are now aware of that low humming or buzzing sound. So that's the main job of the thalamus. It was a relay center, and it was capable of filtering out some information. But it can make a decision as to whether to filter out that information or not. You were ignorant of that buzzing sound for a while until I talked about it, and then suddenly you could hear it. Now, there are some changes to this pathway for different senses. For instance, not all of our senses go to the spine. Vision, hearing, and smell travel up cranial nerves directly to the brain. And not everything goes through the thalamus, but almost everything does. Once again, almost all sensory information gets relayed through the thalamus, and the thalamus gets a chance to filter that information. That's a very important task, because we are getting bombarded with all sorts of information, way more than we could possibly pay attention to all of the time. We have to decide what we're going to focus on and what we're going to ignore. Next up on our list of general concepts is that of referred pain. Not all of our organs have sensory receptors in them to send information to the brain as to what's happening to them. Many internal organs, for instance, have no pain receptors. So when they get injured, they cannot tell the brain directly, and instead this information must be referred. Now, there's a number of theories as to how referred pain works, and nobody's really sure which one is correct, but I'm only going to go over one, the one that I think is best. Injury to an internal organ, like the gallbladder, can cause inflammation, and the damaged gallbladder cells can release inflammatory molecules, which will diffuse to nearby dendrites in the skin of pain neurons. Of course, diffusion does not happen in a straight line, but outwards in a radiating sphere. And so the dendrites out in the skin will be detected over a broad area of skin. And the pain that we detect from gallstones is actually detected by a wide and kind of diffuse region of skin. It's hard to really pinpoint where that injury is. So that's the concept of referred pain. Different internal organs tend to have specific patterns to regions of the skin where that pain is felt. But it's always fairly abstract, difficult to locate, and over a wide area. For instance, the pain of a heart attack is often felt on the left side of the chest, and or all the way down the length of the left arm. That is, of course, if your patient is a man. Heart attacks in women are a lot more complicated. You probably didn't have to come to anatomy class to learn that women are complicated, but the patterns of heart attacks in women can be multitude and various. And for this reason, heart attacks in women have historically been frequently not identified properly. And it's very important to determine when a woman is having a heart attack versus something else. So when you learn about the signs and symptoms of heart attacks and referred pain, make sure that you're learning the patterns for both men and women. The patterns won't be on my test, just the concept of what is referred pain. So those are some of the general concepts that will be true for most, if not all, of the different senses that we're going to be talking about next. We'll need to worry about the difference between sensation and perception. It'll usually take 3 neurons for the information to reach the cortex for us to become consciously aware of the sensory information. Some of our sensory neurons can adapt, meaning they will stop sending action potentials to the brain. That's different from the thalamus deciding whether to relay the information or not. The thalamus can change its mind. Our sensory neurons that adapt do not change their mind. And we also talked about receptive fields and mentioned that a broad receptive field may be more sensitive, but it has lower resolution. Next up, we're going to start talking about some of the senses one at a time. But quickly, before we do, because we're anatomists, we're going to make some lists. We love lists. We can classify the senses and the neurons that detect those senses in a couple of different ways. First off is by the location of the sense that we're picking up. For instance, exteroreceptors are neurons that pick up information from outside of the body, like temperature or sound, whereas interoreceptors tell us about what's happening inside of our body, like blood pressure and bladder pressure. And just to make things confusing, that might seem like enough, we also have proprioceptors. These are special interoreceptors that tell us about the position of our body at any given point in time. These are very important neurons. These are the neurons that prevent us from smashing our teeth together when we chew, or from accidentally poking our eye if we reach up to scratch our eyebrow. We know where these parts of our bodies begin and end, and where they are located, even if you can't see them. But that's not really how I'm going to be teaching the different senses. I have to do them one at a time. So instead, we're going to be focusing a bit more on the types of sensations. And I'll spend a lot of time talking about the sensation of pain and the neurons that detect pain, which are called nociceptors. We'll talk about temperature, mechanical changes, and chemical changes as well. Nociceptors are a special type of neuron, and I say that they are pain neurons, but that's a bit of an exaggeration. Pain is the perception. The sensation that they detect is nociception. And there's going to be a couple of instances where we're going to need to differentiate between the sensation versus the perception. Hang on, my cat wants food. Okay, he's been dealt with. Now we can move back to the sensation of nociception. And what the heck this is. Why don't I just say pain? Nociception is the detection of injury to cells in the body. I could perceive this as pain, but that's not always going to be the case. An injury to the skin, for instance, may simply tear open a bunch of the dendrites of a nociceptor located there. And as those dendrites get torn open, that would allow sodium to rush down its electrochemical gradient and enter the cytoplasm, depolarizing the nociceptor, bringing it to threshold, causing it to fire an action potential, and sending this information to the brain, which the brain could interpret as pain. But you don't have to literally damage the nociceptor for these cells to detect that there's damage to the body somewhere. For instance, the nociceptor may be intact, but if cells nearby are damaged, they will release inflammatory molecules, which will slowly diffuse until they reach the dendrites of the nociceptor and bind to receptor proteins on the dendrites. So these inflammatory molecules behave like neurotransmitters, binding to receptor proteins, triggering graded potentials, and if those graded potentials are strong enough to bring the axon hillock to threshold, this neuron fires an action potential, and it gets sent to the brain, and the brain probably perceives this as pain. We've got two different types of axons that send information to the brain. One is myelinated and the other is not. So some pain signals will reach the brain faster than others. The type A fibers are myelinated, so we call this fast pain, and it'll reach the brain very quickly. This is often prickling or poking or stabbing pain. On the other hand, the type C fibers, of which there are quite a few, this is slow, dull, and aching pain. Pain is tonic, so we frequently don't need a constant update. If you stub your toe, that toe is going to feel painful for a while. So we don't need a quick update every millisecond on whether the toe is still injured. Yes, it's still injured. You are still stupid for walking into that object, whatever it was. You should be more careful next time. Oh, and hey, it still hurts. And yep, still hurts. Yes, still hurts. All right, you got that? It's still hurting? All right, we don't need super fast updates on pain frequently. So a lot of our pain neurons are more slow in their signal propagation. We're going to keep getting the same signal over and over and over. Next is this theory of spinal gating. This was a very good theory, but it didn't turn out to be true, so don't spend too much time trying to learn about the mechanism of this theory. It's the concept that's really important. The theory of spinal gating was that we could change pain signals before they reached the brain. For instance, maybe you hit your knee on something and it hurts really badly, so you rub your knee, and it seems to help. It doesn't quite hurt as much. That obviously happens. I mean, we've all experienced that. So some scientists came up with a theory to try and explain that. And their thought was that some other sensory neurons, like the touch of rubbing your knee, might be able to modify the pain signal before it reached another second-order neuron in the spinal cord to be sent up towards the cortex. And the painful stimulus on its own would be more painful than a painful stimulus plus a touch stimulus. So that was a pretty good theory, and they came up with this map here of maybe how it worked. And it turns out not to be right, and yet we've all done that. We all rub our knee when we get a boo-boo. So what does happen? And it turns out pain does change, just not this way. To understand how pain changes, we need to talk about how it reaches the brain first. And it turns out it's complicated. There's more than one route for pain information to reach the brain. So we've got nociceptors out on our skin. That part is pretty simple. Dendrites in our skin and joints in a few other places can detect damage to the body. Inflammatory molecules activate ion channels. This depolarizes the cell, and the axon fires an action potential, and this information travels to the dorsal horn. It's here that things start to get complicated. The first path is just like I told you about earlier. A second-order neuron would have an axon traveling up one of the ascending columns to reach the thalamus, and the thalamus is probably going to decide that's pretty important. So a third-order neuron could send this information to the post-central gyrus. and your brain would not only be aware that there is pain, but thanks to the homunculus, it would know where that pain is located as well. So that's one of the paths by which this information can reach the brain. But it's not the only one. The nociceptor can bind to a different neuron in the spinal cord, and its axon doesn't travel to the thalamus. Instead, it goes to the reticular activating system. If you were asleep, you're probably not anymore. Now you're awake, and it's from here that this information could also reach the post-central gyrus. We're still not done yet. It turns out this information from the reticular activating system could go to the limbic system, meaning to subconscious parts of our brain. So it's possible that you may not even be aware of a painful stimulus, but it could be affecting your emotions. Or you could be very well aware of the pain and it's affecting your emotions. You're in a bad mood now. So there's a lot of different ways that pain information can reach the brain. And that means that if you want to block pain from reaching the brain, you need to block all of these pathways. Just like you'd have to block multiple roads to prevent somebody from reaching their driveway if there's multiple ways to get to their house. Activating action potentials anywhere along this pathway will be detected as pain. Our brain doesn't really know the difference between whether the dendrites are being stimulated or whether we're activating something halfway up the stream. Conversely, to block pain, or I should really say attenuate pain, it's very difficult to block pain entirely. So the word attenuate just means to lessen the pain, to turn it down. And that's going to be your job, probably, to help turn down the pain in your patients, to help make it manageable. To do that, you're probably going to have to inhibit action potentials in multiple locations. One is not going to be enough. So we've got two different types of pain in the body: nociceptive pain and neuropathic pain. Nociceptive pain is the simpler one. Damage to some part of our body is detected by the dendrites of our nociceptors, and this information is sent up to the brain, where I perceive it as pain. Neuropathic pain, on the other hand, is when signals originate somewhere else, like in the spinal cord. Perhaps I have a slipped disc that is pushing on the spinal column, and that's forcing some voltage-gated sodium channels open. And when they open up the ones nearby, which open up the ones nearby, and so on and so forth, until this information reaches my cortex, and it feels like my entire leg is in pain. And that's because my brain doesn't know that the signal was starting halfway up. It's always going to assume that the signal came from where the dendrites are located. To treat nociceptive pain, we can use drugs that block the receptor proteins on the dendrites. NSAIDs like aspirin and opiates are really good at doing this. Those don't work very well for neuropathic pain. That's because dendrites aren't really involved. So to treat pain that arises from within the nervous system, we're going to have to use drugs that mess with action potentials. For instance, anti-seizure medications like gabapentin might be used to treat neuropathic pain, whereas morphine would be better at treating a really bad broken leg. Let's go back to that spinal gating theory, the idea that we could change how painful a pain signal was. And while they got the mechanism wrong, the idea was absolutely right. And it turns out that activation of the limbic system can activate neurons that travel down to the spinal cord and cause EPSPs or IPSPs on some of these second-order neurons, making them more sensitive or less sensitive. Opiates, for instance, can be released, causing IPSPs, making these second-order neurons less sensitive. And for that reason, the painful stimulus happening out here may not be transmitted when it reaches the spine, and your brain doesn't feel it. Or, more realistically, it feels less painful. Conversely, if you're in a really crappy mood, the limbic system could also fire EPSPs down here, making things that shouldn't feel painful actually feel painful. So our limbic system, or our emotions, really can affect how much pain that we feel. And we know the mechanism for this. There are neurons that we have mapped out that account for this behavior. Okay, you got that? If not, hit the backwards button, because it's about to get even more complicated. It turns out that activating a nociceptor usually isn't enough to cause the perception of pain. In fact, we rarely perceive things based off of just one of our senses. This will be really obvious when it comes to taste. We rely on our sense of smell actually a lot more than our senses coming from the taste buds of our tongue. And pain is no different. To feel something painful, we usually need another one of our senses to corroborate that information. And vision is a very powerful one. I'm sure you've all experienced this. A small child falls down and skins her knees, and she stands back up and seems fine until she looks at her knees and sees that they are bleeding, and suddenly she starts crying. It wasn't until her visual sensory systems matched her nociceptive systems that her brain actually perceived both of those pieces of information as pain. Prior to that, the nociceptors were just sending nociception, and her brain may have been aware of it, but was not perceiving it as pain. If you can distract that small child, oh my goodness, look at the ground, what did you do? Did you hurt the floor? If you can distract that child, it might attenuate the pain, get her vision onto something else other than her bleeding knees. So there's a high degree of overlap between pain and vision. And that's very important for us. The things that we see as painful will feel more painful. And that doesn't just exist to the individual. Now, what do I mean by that? What I mean is that if you see somebody else experiencing pain, it can feel kind of painful to you as well. And I'm sure you've experienced that as well. You see somebody getting stuck with a needle, or you see an operation happening. The same parts of your brain that are registering that visual information are also responsible for the pain information, and you may be experiencing that as the sensation of pain, not just vision. This makes us good social animals. For many of you, one of your main jobs is going to be managing pain in your patients. And one of the big parts of that is going to be explaining to them that you can't stop the pain. You can only attenuate the pain. Maybe you're not going to use that word. One of the problems is that we have no way of measuring pain. I don't have a painometer that I can simply stick into my patient and go, oh, you're at pain level 7. Ah, you're only at pain level 4. Shut up and quit being a baby. We just have to ask our patients how much pain that they are in. And the problem with that is that they don't know. Depending on how much pain they've been in their life, the answer that you get could be very different. So the best that we can do is this Wong-Baker pain scale, where you're going to ask your patients to estimate the amount of pain that they are in. And in theory, the only time that they should give you the answer of 10 is if they were on fire and you were stabbing them with multiple scalpels at the same time. If that's not happening, they should be giving you a number lower than that. For many hospitals, if patients give you a number even higher than 10 out of 10, more than the maximum amount of pain possible, you're going to treat that as a 0, and you're going to leave and not give them any pain medication at all, until they learn to play the game properly. Of course, one of my favorites over here from Gomer blog, which if you're not familiar with, go check it out, is the pain in my ass rating scale, very similar to the Wong-Baker scale. And that goes from 0 to where, you don't have to worry about your patient, to if they call you 10 times every hour, you transfer them to somebody you don't like. Many pain medications can be centrally addictive, especially the opiates. And for that reason, we really shouldn't dish out opiates like they were candy. And so if your patients are in pain, you're going to try to start them out with a weak pain drug, a non-opiate, like ibuprofen or aspirin. And if that doesn't do the job, you might move them up to one of the weaker opiates. And only if that doesn't work would you move them up to one of the stronger opiates. This is called the pain ladder. And with no other information, you should follow this ladder. Of course, if your patient is missing an arm, you're not going to be starting them off on the aspirin. You can move them up to something stronger. Like I said, in the absence of any other information, stick to the pain ladder. And another thing that you should stick to is a schedule. It turns out that addiction is like learning. And just like our dog, learning that peeing in the house is bad, the way to train the dog not to pee in the house is to reward the dog for peeing outside at the same time. It's the two things happening at the same time that we can learn very easily. And your patients are going to learn very quickly that they love drugs when they ask for drugs and they get them. They're going to learn a behavior, a drug-seeking behavior. Well, if you don't give them the chance to do that, if you only give them these addictive drugs on a schedule, that takes away that drug-seeking learned behavior. It doesn't make opiates non-addictive, but it'll decrease the chances that your patients build up drug-seeking behaviors. Next up, and this is just for fun, but there are different types of drugs that can attenuate pain. The ones on the left are called painkillers or analgesics, and these include some of our favorites like aspirin and other non-steroidal anti-inflammatory drugs. These typically block the production of inflammatory molecules. The opiates are another class of drug. You may have inflammatory molecules binding to dendrites, but if you've got opiates producing iPSPs on this neuron, and the inflammatory molecules are creating EPSPs, you might be able to stop that neuron from firing an action potential. And we're learning a bit more about the cannabinoids. It's difficult because these are still regulated as a Schedule I drug by the federal government, which means that researchers can't do research on them without jumping through hundreds of very difficult hoops. But maybe that'll change sometime soon, and we'll be able to learn more because there's a lot of promise here with these chemicals. On the other hand, anesthetics are chemicals that can block action potentials. They don't stop things happening at the dendrites, so the axon may reach threshold, but you can't open voltage-gated sodium channels, and so the information isn't sent to the brain. There can be local anesthetics, or inhalants are pretty famous because these were the first painkillers. And there's some very interesting medical history as to the difference between ether and nitrous oxide for the use of blocking pain. Both opiates and cannabinoids work in the human body by mimicking natural neurotransmitters. There's a lot more understood about opiates because the federal government believes that these are less harmful to human health. So that's what I'm going to talk about. But the same would be true, just with different names, for the cannabinoids. These chemicals mimic natural neurotransmitters. The opiates mimic endogenous opiates, which can bind to one of 3 different types of receptor proteins. Remember the adrenergic receptors came in the alpha and beta flavors? Well, the opiate receptors come in the mu, kappa, and delta flavors. But like the adrenergic receptors, these are just a different type of G protein coupled receptor. Morphine, our classic opiate, one that has existed for at least as long as written language has, and probably even longer, just nobody could write down to tell everybody else how wonderful poppy extract was. This type of chemical activates the mu opiate receptor. And on the dendrites in the spinal cord, this causes IPSPs, which blocks pain information happening out in our body from actually reaching the brain to be perceived. There are opiate receptors in a lot of different places besides the spinal cord. For instance, there's a number of them in the digestive tract doing other stuff. And for that reason, morphine can block the movement of food through the GI tract, causing constipation and loss of appetite. The opiate receptors are also found in the nucleus accumbens. And here this causes activation, which leads to reward and euphoria. And this makes the opiates, like morphine, centrally addictive. These are one of our classic addictive drugs. For that reason, drug companies have been looking for non-addictive painkillers for a long time, because this is a major came up with oxycodone, which was marketed as a non-addictive opiate, because it tends to activate the kappa receptors, which does the exact opposite of the mu. It tends to inhibit the nucleus accumbens. And for this reason, they were marketed as non-addictive. Well, we really should have done our homework, because 100 years ago, this chemical had been discovered and everybody knew that it was addictive back then. And so when it was rebranded as non-addictive, well, we all looked and saw what we wanted to see. Ooh, it activates kappa, not mu? Well, then it shouldn't be addictive. Let's start prescribing this like gangbusters. And for reasons that we still don't fully understand, oxycodone is centrally addictive, just like morphine is, even though it acts through a different type of opiate receptor. For that reason, opiates aren't very good, and some would say very bad, at treating chronic pain. They're good for acute pain. Somebody's been in a car crash, they've broken a leg. If you think that that part of their body is eventually going to heal up, giving them an opiate is going to be a really good way to manage their pain as they go through the healing process. But for chronic pain, like neuropathic pain, for somebody who has a bulging disc pushing on their spinal cord, the use of opiates is not going to work very well. It may block the pain for a while, but people build up a tolerance to the opiates because they down-regulate their opiate receptors. And so the same amount of drug has less of an effect a few weeks later. And because it also activates the nucleus accumbens, they may have to start taking that drug simply to avoid withdrawal symptoms, rather than taking that drug to avoid the pain due to the injury of their body. Ideally, we would like to fix the problem. If it's a bulging disc, go through surgery so that it's no longer pushing on the spinal cord, and eventually the pain will go away. But we don't always have that option. We don't always know what's causing the chronic pain in our patients. And if some of the other pain medications, like the neuroleptics, aren't working, then sometimes opiates, while not being a very good solution, are the best solution that we have, even if it's a crappy one. And this is a big deal right now in bioethics. A lot of states are clamping down on the prescription of opiates for pain. And in some places, people with chronic pain conditions can't get the drugs that they want. Now, that might be in their best interest, but it might not. And we're not really sure. It's hard to make a law that's going to be the right one for everybody. There are a lot of people who are going without the best possible pain medications. And even though the opiates aren't very good for the treatment of chronic pain, until we really develop better ones, they might be the only ones out there for somebody. And it's important to not make new addicts. We don't want people becoming addicted to drugs because then they can't have families or operate in society. But of course, if somebody's in horrible pain, they also can't be a productive society member or family member either. So we don't always have the ideal solution. But that's important right now. Heck, maybe by the time you're watching this, the problem has been solved, and so I should really just move on. Moving on in chronic pain management, my prediction is that we're going to have to pay a lot more to the cannabinoids and some other chemicals that are released by glia. It turns out that pain signals, for instance, being transmitted from the second order to the third order neuron, can be modulated by the activity of glia, like astrocytes. These cells can release other chemicals that act as neuromodulators, making the postsynaptic cell more sensitive. They actually make you more sensitive to pain. And so maybe we shouldn't be designing drugs that affect neurons. Maybe we should spend more time designing drugs that affect the activity of the glia. For instance, one of those drugs is called Sativex. It mimics a cannabinoid. THC, one of the active components of marijuana, can activate all sorts of cannabinoid receptors, whereas Sativex only activates one of them, meaning it can attenuate pain. It has the same pain-lowering properties as smoking or eating marijuana without having the same central effects. It doesn't have the euphoria, for instance, or the change to appetite. So that wraps up pain, and I spent a heck of a lot of time on it because it's the most medically relevant of our general senses. But that means I'm not going to have much time to spend on the rest of the general senses. The next of our general senses were the thermoreceptors. These are neurons that can detect changes in temperature. Right now, you're probably sitting in a room that feels like it's room temperature. If you were to move to some other location, you might notice that the temperature was different, but after a while it would start to seem, just the temperature. And that's because thermoreceptors are phasic, meaning they adapt very well to signals that don't change. Our thermoreceptors only alert us to when things are warmer or colder. They do not tell us the exact temperature at any given time. Next up are the mechanoreceptors. These are neurons that can convert physical information into action potentials or electrical information. Those physical changes may include stretching, pressure, twisting, or other physical distortions to membranes like the skin. There are a number of different types of mechanoreceptors. There are a number of tactile receptors in the skin, but there are also the baroreceptors, which can detect changes in pressure. We already talked about some baroreceptors when we discussed the baroreflex. Changes in blood pressure in the aorta and carotid bodies were sent to the brain, and that could alter activity of the sympathetic and parasympathetic neurons back to the heart. There are other types of pressure that we might be interested in, such as the pressure in the lungs or the bladder. And proprioceptors are also mechanoreceptors. They detect the amount of stretch that happens in tendons and muscles. And all of this information can be put together in the cerebellum so that we have an idea of where our body is located in space at any given time. Even if we're not looking at what our arms and legs are doing, we feel what they are doing. Of the mechanoreceptors in the skin, there are fine touch, fine pressure, crude touch, and crude pressure receptors that give us different types of touch information. These can be further characterized by their shape and the people that discovered them and named these neurons after themselves, like the Ruffini corpuscles and Merkel discs. I'm not going to spend much time on this. This is something that you might have done in the lab section had I thought it was important. Baroreceptors are going to be more important this term because blood pressure is an important topic this term. These respond pretty quickly and adapt pretty quickly as well. Proprioreceptors are found in our tendons and muscles and a few other places, and most of this information is sent to the cerebellum as well as the cortex. My cerebellum needs to know what my body is doing at any given time if it's going to be able to manage balance and posture and allow me to walk without having to think about it too much. For instance, if you close your eyes and touch your fingers to your nose, you've just used a bunch of proprioceptors to help guide your finger towards your nose, even if you weren't looking at it. Chemoreceptors are a large class of receptor proteins, or the cells that have those proteins, that detect certain chemicals. So these chemicals would be very similar to a neurotransmitter, but instead of chemicals released from synaptic terminals, these are chemicals just found in our blood or floating in our nasal cavity. Some common chemoreceptors include those that detect concentrations of oxygen, carbon dioxide, or glucose. So those are the other general sensory receptors. Again, I spent most of our time focusing on the nociceptors, which were the type of cell that were activated when we detected pain. Pain, of course, being the perception. But I had other cell types that could detect other types of information. The thermoreceptors for temperature, a large number of mechanoreceptors for different physical information, and the chemoreceptors, which could detect a specific type of chemical. But that's going to wrap us up for the general senses. And again, we covered a number of basic concepts that were common amongst both the general senses, but are also going to be common amongst some of the special senses that we're going to be covering next. The special senses are those that require extra organs to help the dendrites of the receptor cells to convert information from outside of the body into action potentials. Our special senses include smell or olfaction, taste or gustation, vision, equilibrium, and hearing. Let's start with the sense of smell. Technically, smell is the perception, whereas olfaction is what happens in the nasal cavity, where chemicals floating in the air can bind to cells and trigger action potentials. Those action potentials may be interpreted in the brain as smell, but the conversion of chemical signals in the nasal cavity into action potentials in neurons from cranial nerve 1, that is olfaction. So the dendrites of these neurons extend into the mucous membrane of the olfactory epithelium. These olfactory receptors are very special types of neurons. One thing that's very special about them is that their dendrites are right there at the surface of the body, meaning they're going to come into contact with a lot of damage. We inhale a lot of stuff all day long that can kill cells. Because most of those cells are epithelial and are rapidly dividing, that's no problem. I don't think about that damage. It just happens and the cells get replaced. But the dendrites to the neurons, that's harder to replace. And if you were to kill the neuron, well, we said a few weeks ago that neurons don't divide. I wouldn't be able to get it back. So the olfactory receptors are going to be very special. When their dendrites detect information, this will be converted into action potentials that travel up cranial nerve 1. This starts at the olfactory bulbs, which kind of sit there down on the ethmoid bone on the cribriform plate, and they travel all the way back to the brain. This is one of the type of senses that does not actually go through the thalamus. One of the others was pain. Pain could go through the thalamus, but it didn't have to. There were alternate pathways that went around the thalamus, and smell is another one. In order to smell something, you first have to be aware that there are chemicals floating around in the air. And the olfactory neurons in your olfactory epithelium have to convert those chemical signals into electrical signals. But this is very similar to what we learned about in Chapter 12. Instead of neurotransmitters binding to neurotransmitter receptors on the dendrites of neurons, now we've just got chemicals floating in the air, binding to receptor proteins on the dendrites of the olfactory receptors. These proteins are one of a large family of G protein coupled receptors. There are hundreds of different odorant receptors, each of which has a slightly different active site, capable of binding to one or more specific odorant molecules, chemicals that our nose can actually pick up. Not every chemical has an odor, meaning I don't have receptor proteins that can bind to carbon dioxide or oxygen in my olfactory epithelium. And for that reason, oxygen and carbon dioxide don't have any smell, but other things do. And that's because those chemicals bind to not ligand-gated ion channels. They bind to G protein coupled receptors. And that's actually very important. We want to be able to detect very small quantities of a chemical in the air. And second messenger-linked receptors are good because they amplify signals. And that's something we definitely need to do when it comes to the sense of smell. I mentioned earlier that the dendrites to these olfactory receptors extend all the way out to the surface of the olfactory epithelium. And for that reason, they come into contact with a lot of toxins and poisons. Smog, pollution, and other nasty stuff can damage cells. There's a lot of epithelial cells that get replaced there all of the time. And neurons can grow new dendrites if their old ones get damaged. But with enough damage, those neurons might actually die. And it turns out that does happen in our olfactory epithelium. So the neurons here do something pretty darn special. They go through mitosis, which is not very common for neurons throughout the rest of the nervous system. In fact, we said earlier that neurons don't do that, with a few exceptions. One of the other exceptions is shown to the picture on the right. Neurons actually do this in the hippocampus as well. We're not entirely sure what the neurons are doing dividing in the hippocampus. But if you inhibit this cell division, it tends to lead to diseases like depression. So we should probably be studying how the neurons of the olfactory epithelium can undergo mitosis. When other neurons don't do this, or when they do and they don't do it enough, it leads to diseases like depression. So this is a very important concept. It's already put into application once. A few years ago, a man who suffered full paralysis, and the doctors were absolutely sure that the spinal cord had completely degenerated at this spot. They took some of the neurons from his olfactory epithelium, removed them, and injected them into the area of his spine that was damaged. And it turns out those neurons were able to divide. And rather than becoming olfactory receptors, they realized they were in the spinal cord, and they became spinal neurons. And this man was able to regain some motor control over his body, and in fact has been learning to walk with a lot of support. But that's 100% different from what we actually do in medicine. Right now, if one of your patients become paralyzed, you have to tell them that's going to be permanent. There is no way to reverse that. But this one experimental treatment was able to reverse that spinal paralysis. And that represents a huge change in how we think about damage to the central nervous system. This was done in Poland, and it was only one patient, so we have to hold our breath a little bit and see if this can be repeated. But it turns out similar experiments were done in the United States, not by taking neurons out of the nasal cavity, but by simply electrically stimulating the spinal cord. That seemed to be able to induce some growth and cell division as well. How? I don't know, but again, I think the important part is not so much how it works, just that it can work. So pay attention to this in the future. Again, this is not currently medicine. This is still experimental science at this point, so I can't promise that this will actually hold true. Right now, it's just promising. Next, let's move on to gustation. This is what happens when chemicals bind to taste receptors on the tongue. This is a big part of taste, but taste is a perception that occurs up in the brain, and like pain is going to involve more than one of our sensory systems. The sensory system on the tongue, or at least the one localized to taste buds, is called gustation. Within a taste bud, we find neurons which can send signals to the CNS, as well as some extra epithelial cells which help these neurons to convert chemical signals in our food and saliva into electrochemical signals that we learned about in Chapter 12. Taste buds, like our olfactory neurons, are exposed to a lot of toxic chemicals. There's a lot of things in food that you may not think of toxic. They're not going to kill you, but they do kill some of your cells. So it's important for these cells to regenerate. And like any good epithelial cell, there are some stem cells which can divide and differentiate into the actual sensory cells, which can connect to neurons that will send the information to the CNS. Now, recall when I was talking about smell. Wait, you don't remember teaching you about smell? Hit pause, rewind, go back to that section. It's only about 20 slides ago. Okay, now that you're back. Smell had hundreds of different G protein coupled receptors, proteins on the cell surface of dendrites whose active sites could bind to different chemicals floating around in the air, and this could activate action potentials in the neurons that I detected as the sense of smell. On the tongue, I only have 6 different gustatory receptors. The 4 on the left, sweet, salty, sour, and bitter, were the ones that I learned about in school. Since then, 2 more have been discovered. There are water receptors that can detect when I drink water, and another type called umami. This is the taste of glutamate, which is an amino acid. And typically, we find a lot of glutamate in proteins, but it's bound to other molecules. So we only taste this molecule when the protein has broken down somehow. For instance, if you cook beef or chicken in certain ways, or Parmesan cheese, or soy sauce, you may detect a lot of glutamate molecules in those foods. These chemicals can bind either to a ligand-gated ion channel or to a G protein coupled receptor. Let me talk about a ligand-gated ion channel that can bind to sour molecules. Luckily, these receptors are a bit promiscuous. They will bind to a number of different ligands. I don't need hundreds of different receptor proteins for hundreds of different receptors. I just need to be able to bind to a part of that molecule, a part of that taste. Sour chemicals can bind to ligand-gated ion channels, opening them up, allowing sodium to rush into the cell, depolarizing it, and if it brings that cell to threshold, it fires an action potential and sends that information to the brain, where I can detect that as a sour taste. If you add some sodium to that food, well, now there's more sodium in the saliva. So when that same sour molecule binds to the same ligand-gated ion channel, now there's a bigger electrochemical gradient. Even more sodium is going to rush in. I'm going to fire a higher frequency of action potentials, and this food is now going to taste even more sour. Now, if I had enough sodium, it could bind to salty channels. But in this case, sodium is simply acting as a flavor enhancer. In fact, we add salt to many foods that we make, not so that it tastes salty, but so that it enhances other flavors within that food. Now, before I move on, for any of you food nerds out there, there's more than one way that salt can do that. For instance, a salt rub on meat can help pull water out of the meat because it's a solute. And when that happens, it pulls some of the other molecules to the surface of your meat, making it more flavorful. But in this instance here, salt acts as a flavor enhancer simply because salt is sodium chloride, and sodium is one of the major electrolytes that we use in our ion channels when we activate neurons. There's a lot more to taste than simply activating taste buds on the tongue. There are other neurons on the tongue which can detect signals as well. For instance, some foods contain chemicals, such as capsaicin found in peppers, which can bind to and activate nociceptors. But when these nociceptors send signals to the brain, they are usually interpreted as spiciness. That's the perception, not pain. These chemicals can do damage to cells, so it's important for us to be able to detect them. A small amount of them can actually be tasty, but too much can cause an adverse reaction. We need to avoid too much capsaicin. In fact, capsaicin can cause damage to the dendrites of these nociceptors, causing them to regress. And with fewer dendrites, these nociceptors are less sensitive. And if you're less sensitive to capsaicin, that means you have a high tolerance for spicy food. That's okay. Avoid spicy foods for a month or two, and those dendrites will grow back, and you'll have lost your tolerance. There are other neurons on the tongue that are important for flavor. For instance, temperature and touch neurons send very important signals to the brain, which we interpret as taste. Foods should be the right temperature and the right texture, otherwise they might not taste too good. The 6 types of taste buds are not distributed evenly across the tongue, but tend to form clusters. We cannot, however, make a map that would be consistent from person to person of where these taste buds are located. It's fairly random. So if you have an older textbook that tries to tell you about the taste map on the tongue, it's wrong, and it's always been wrong. And yet many of us have learned about this in school, and it's time for this myth to be demolished. This just goes to show that a lot of textbook authors simply look to the previous textbook to determine what should go into their textbook, rather than actually trying to determine if what they're saying is true. We've seen a pattern here that's going to be true for the next couple of hours. That is that the perception that occurs up in the brain actually relies on more than one of our senses. The perception of taste relies not only on gustation, which occurs on the tongue, but also activation of nociceptors, temperature and touch receptors. And a large part of taste involves activation of the olfactory epithelium in the nasal cavity. And I didn't even go into memory. That's a big part of taste as well. If you got sick from a food once, you're probably not going to like the flavor of that food ever again. That memory alters how it tastes. It doesn't change the activation of taste buds on the tongue, but it changes how you perceive that information. So this would be a good spot to take a break, grab some food, get something to drink, think about gustation and olfaction, and I'll meet you when you're done. Okay, welcome back. In this second part of the lecture, We're going to learn about the ear, and I'll discuss talking to Ralph on the big white phone, and why it is that when we get dizzy, that sometimes makes us want to vomit. We'll talk about a number of different types of vertigo that will help explain different aspects of the physiology of the inner ear. We can also ask the question, are you able to make your ears click, producing a sound that only you can hear just by wiggling them a little bit? If you can, that means you have cortical control of a couple of tiny skeletal muscles in your middle ear. And if you have no idea what I'm talking about, then you can't control those muscles, and the rest of us are just better than you. We'll talk about how we understand speech and music. To do that, we'll actually have to go into a little bit of physics. We'll talk about high-frequency hearing loss and why that's so important to our ability to understand speech, but also our ability to locate where sounds are coming from. And lastly, we'll talk about cochlear implants, which I think are a cool example of bionics, using machines to enhance human abilities. To do all of that, we'll start by discussing the outer ear, move our way to the middle ear, and then to the inner ear. And as we do so, we'll have to learn about a few diseases which help explain the physiology that we're discussing, and we'll even have to go into a tiny bit of physics to truly understand how the inner ear works. Let's start with the auricle, which is what most people would call their ear. These are structures that help with localizing sound, so we'll come back to this when I talk a bit about physiology. This will open up to the external acoustic meatus, inside of which there are a number of ceruminous glands. These are modified sweat glands that produce a waxy material, which helps to trap microorganisms and push them outside of the body. The external acoustic meatus terminates in the tympanic membrane. This structure is very important for hearing. It does the opposite of what a drum head does. And this is where it gets its name, tympanic or timpani. If you were to smack a drum with a drumstick, the head of the drum would vibrate and it would produce a sound. The tympanic membrane does the opposite. Sounds cause it to vibrate. If it can't vibrate, then it can't transmit sounds to the middle ear, and this leads to problems with hearing. Damage to the tympanic membrane in the form of a rip or a tear, or even pressure on this membrane caused by inflammation, can prevent it from vibrating properly. For instance, a middle ear infection may cause fluid to build up behind the tympanic membrane, putting pressure on it and limiting its ability to move. And when this happens, it will limit our ability to hear sounds. To relieve this, an ear tube can be inserted into the tympanic membrane, allowing fluid in the middle ear to drain out the external ear. Here's a picture of an ear tube. One of the other nice things about an ear tube is that if that middle ear infection were to get so bad that it might cause a tear in the tympanic membrane, a small incision and the insertion of a tube will be much easier to heal over. This will lead us to the next region, which is the middle ear. Inside of this chamber, you can find three small bones, or the auditory ossicles, called the malleus, the incus, and the stapes. These bones are going to transmit sound waves from the external ear to the internal ear. Vibrations of the tympanic membrane will cause the auditory ossicles to move, which will transmit the vibrations to the fluid of the inner ear. Because the auditory ossicles are connected to each other at angles, they behave like levers, which can amplify the sound as it reaches the inner ear. If the sound waves are already loud enough, there are two skeletal muscles here in the middle ear which can attenuate or decrease the volume of the sound. These include the tensor tympani muscle and the stapedius muscle. If you've ever gone to a loud concert, and after a few minutes it doesn't seem as loud as it did when you first walked in the door, that's activity of these muscles dampening the sound waves before they reach the neurons found in the cochlea. Some of us can control these skeletal muscles consciously, which cause these bones to move, which will cause the fluid in the inner ear to move as well, which we interpret as a sound. It sounds like a click or a thump. If you can't do this, you must just be in awe of those of us who can, right? The inner ear is a series of fluid-filled tubes that are all continuous with one another, that float inside another series of fluid-filled chambers. The vestibule is the middle section. Connected to that are 3 semicircular canals on one side and the snail-shaped cochlea to the other side. Let's start with equilibrium. This is going to be detected by movements in the fluid picked up by specialized cells called hair cells. These cells contain stereocilia, which are not cilia at all but microvilli, and a single large kinocilium. These structures can bend when pushed on by fluid, and we will interpret this as movement. These hair cells are located in 3 spots within the inner ear. Rotational information can be detected by hair cells found in the ampullae of the semicircular canals. Acceleration and tilting can be detected by hair cells found in the vestibule. We also have hair cells in the cochlea, and we'll come to those in a moment. The hair cells found in the semicircular canals are able to convert rotational movements into action potentials, and these are found at each of the 3 wide ends of the semicircular canals. There are 3 semicircular canals oriented in the X, Y, and Z axes, and within these canals are fluid. This fluid tends to have its own momentum, so that when the body rotates, the fluid tends to stay in place for a little while. And when we stop rotating, the fluid continues to move. It's the movement of this fluid pushing on hair cells that will allow us to interpret this as rotation. The hair cells in the vestibule get a little help from otoliths. Literally, that translates as ear stones. So there are tiny rocks within the fluid, and these rocks tend to have momentum, just like the fluid does. When we speed up, the rock will be a little bit slower to accelerate. When this happens, those little rocks will push on the hair cells, either in a forward or a backward direction. And that can either open up ion channels or close up ion channels, as the stereocilia are getting pushed over or pushed away from openings in the plasma membrane. These hair cells will be firing action potentials at a base rate. They have tone. But when we speed up or tilt, that will open up some ion channels and depolarize the cell, causing it to fire action potentials at a higher frequency. Moving in the opposite direction tends to close up those ion channels, which will hyperpolarize the cell, lowering the frequency of action potentials. This information would be perceived by the brain as either tilting or acceleration. So, tilting and acceleration are detected in the vestibule. We can further subdivide the vestibule into the utricle and the saccule. And those otoliths actually contain both a gelatinous material as well as a crystalline material. For purposes of your exam, otolith will be enough. To summarize, rotation is detected in the 3 semicircular canals, while tilting and acceleration are detected within the vestibule. The saccule is the lower part of the vestibule, and the utricle is up higher. And some folks have come up with a little mnemonic, "Your sac is below you," to help remember which is which. The letter U here standing for utricle, and the sac, well, if you're not sure what sac we're jokingly referring to in the mnemonic, then I'm not sure I can help you. Rotational information from the semicircular canals can be sent to the brain in a reflex arc that will control the external eye muscles. if we rotate in this direction, this reflex will cause the eyes to rotate in the exact opposite direction. Try it out. Look in the mirror and turn your head quickly. You'll notice you can stay focused at your own image in the mirror, and you didn't have to think about how fast you had to move your eyes to stay focused on that single object. This was carried out by this reflex. We detect the exact amount of rotation, and this automatically causes the eye muscles to move the eyes in the exact opposite direction at the same speed to compensate. If the semicircular canals are sending erroneous information to this reflex arc, or for any other reason signals are being generated along the pathway, this can cause the condition nystagmus, where the eyes move to compensate for a signal that shouldn't be there. When the eyes reach the borders, they then hop back, and they keep getting information from the semicircular canals that the head is rotating, so the eyes try to compensate again, and then they jump back, and they compensate and jump back and over and over. This condition can be temporary. For instance, some good homework might be to drink enough alcohol until you give yourself a little nystagmus. Just make sure you're doing that at the safety of your own home, or you have a designated driver. Some people can inherit this condition. If you don't drink alcohol, another way to induce this condition is simply to spin around 20 or 30 times. Then, when your head stays still, you'll notice that your eyes are still trying to compensate for motion that is not happening. The fluid in your semicircular canals is still moving around. It has its own momentum. So it's pushing on the hair cells, even though your head is now staying put. But your brain interprets this as rotation, and the reflex will try and get your eyes to compensate. So that's another way you could induce a little bit of temporary nystagmus to see what that condition is like, and just be thankful that it'll eventually go away. So if nystagmus was involuntary motion of the eyes, vertigo is the perception of the body being in motion when it's not, that could lead to nystagmus. For some people, vertigo can be triggered when they are up in high places, which can cause them to become dizzy and lose their balance, and for that reason they may have a fear of heights. A lot of people get these two things confused, though. Acrophobia is the fear of heights. Vertigo is the sensation that the world is spinning even though it's not. Typically, our rotational information is sent to the brain from the semicircular canals. But when we are spinning, there's actually more going on than simply fluid moving in the inner ear. Our vision may also tell us that we are spinning around, and our muscles could even alert us to this condition as well. So when our brain determines whether we are rotating or not, it usually is getting information from the semicircular canals, from the eyes, and also from a bunch of muscles. And hopefully all of this information should match. When it doesn't, that's when vertigo can occur. When our sensations do not match, then we get altered perceptions. For instance, if you look at this image over here, your eyes are telling you that something's moving, even though your inner ear is saying that, nope, everything's staying put. And it may make you feel a little bit queasy. Your perception here is a little bit altered. It's inducing a little bit of vertigo. Cervical vertigo will illustrate how the brain often uses multiple sensations before deriving a perception. Under normal circumstances, when we tilt our head, the utricle will be detecting that tilting thanks to the otoliths and hair cells, and it will send that information to the brain. But the brain also receives information from the proprioceptors in the neck muscles. If we tilt our head to one side, one muscle should be contracting, while the muscle on the opposite side should be stretching. And if everything is healthy, this information will match. We'll get a signal that we're tilting to this side from the inner ear, and that we're tilting to this side from the neck muscles. Of course, abnormal tension in these neck muscles may accidentally trigger these proprioceptors. If somebody has a sore neck, those proprioceptors might be triggering a contraction and sending that information to the brain saying the head is tilting, even though it's not. And when the inner ear says, no, we're not tilting, those two sensations don't match. And this can trigger vertigo, or an abnormal feeling that the world is spinning, even though it's not. This type of vertigo can be treated with massage. Another type of vertigo is illustrated to the video here on the right. This is called BPPV, and this can often be triggered by car crashes, which can dislodge one of the otoliths from the vestibule, and it winds up in one of the semicircular canals. And when it's there, it causes a bit of turbulence in the fluid flowing through the canal, which pushes on the hair cells abnormally and can induce vertigo. The brain thinks that it's rotating even though it's not. The treatment for this type of vertigo is fairly simple. We're going to have our patient lay on a table and move in 3 very specific directions. We're going to have our patient move their head in the x-axis, the y-axis, and the z-axis to dislodge that little ear stone and get it out of that semicircular canal. So we'll have them lay on the table, turn their head from one side to the other side, shift their body, and next they should be sitting upright. And at every one of these turns, we should be pushing on that ear stone, getting it to drop out of the semicircular canal. Here in this part of the video, it's showing that by turning the head in these specific directions, we are tilting the semicircular canals so that rock is always falling to the bottom. And by changing where the bottom is located, we should eventually get this little stone to drop out of the semicircular canal, getting rid of the cause of this vertigo. Now that doesn't get rid of this ear stone altogether. It could wind back in one of the semicircular canals just as easily as it got there in the first place. So this treatment might need to be repeated. Luckily, it's very non-invasive and inexpensive. Before I move on, I just want to say that this type of vertigo, I should say, this type of vertigo is often caused after a car crash or other trauma that can break one of those otoliths off. The reason it can get to the semicircular canals from the vestibule is that the fluid-filled chambers of all of these 3 sections of the inner ear are connected to one another. Another way to induce vertigo is to change the density of the fluid in these tubes. Alcohol can do this. It does not have the same density as water, and because people can drink enough alcohol that it has a significant percentage in the bloodstream, it then becomes part of this lymph and will cause one of the fluid-filled tubes to float a little bit higher than it should. This will also push on hair cells, which will send this information to the brain, and the brain will erroneously perceive this as motion, even though the body is not in motion. So this is ethanol-induced vertigo. And this is going to lead us to our next question. Why do we tend to get sick to our stomach when we get dizzy or get the spins? The reason for this, evolutionarily speaking, is that prior to the invention of farming, the only way that humans would have come into contact with large quantities of alcohol was to have eaten large quantities of rotten fruits or berries. And while we might enjoy the effects of a little bit of ethanol, sometimes that leads to food poisoning. So the brain has developed a mechanism to protect us, and that is when it detects signs of ethanol toxicity, meaning when we start to feel like the world is spinning, even though it's not, the brain assumes that's because you ate a bunch of rotten fruits and berries. And so this will induce a bunch of vomiting to try and get rid of the source of what it assumes is causing the vertigo. Unfortunately for people with BPPV or cervical vertigo, the brain does the exact same thing. It'll respond by giving you an upset stomach, even though the cause might be sore muscles in the neck. So those were the first two functions of the inner ear, to detect rotation and to detect acceleration and tilting. Next, we'll move on to the cochlea. Next, we need to discuss how the inner ear can convert sound waves into action potentials. And in order to do that, we'll need to cover a little bit of the basics of the physics of sound. Right away, though, we've got a problem. Neurons can fire action potentials, but the only thing that's going to change with those action potentials is their frequency. And sound will not only have a frequency of the waveforms, it'll also have a volume. So we've got two bits of information that we need to code for with only one thing that can change. We can only change the frequency of action potentials. One thing that's interesting about the ears, however, is that unlike the eyes, we can detect multiple sounds all at once without them blending together to form new sounds. A chord on a guitar, for instance, people can discern the individual notes. Our eyes, on the other hand, when they are bombarded with multiple wavelengths of light, generates new colors. For instance, yellow plus blue equals green. In the ear, we must first get these sound waves to the inner ear. We'll have vibrations in the air, ultimately traveling up the external acoustic meatus to cause the tympanic membrane to vibrate, which causes the auditory ossicles to vibrate, which will cause waves in the lymph inside of the inner ear that will ultimately stimulate hair cells, very similar to the way that we detected rotation and equilibrium. And this will be translated or transduced into action potentials, which will travel to the brain up cranial nerve 8 to be interpreted as sound. First off, the auditory ossicles are arranged in such a way that they can amplify the sound waves. They act as tiny levers. But when they vibrate, they will cause waves in the fluid of the inner ear, just like there are waves on the beach or waves in a swimming pool after you jump in. The stapes bone connects to the oval window, and when it pushes on the membrane of the oval window, that will cause the lymph to generate waves inside the cochlea. These waveforms will travel up the cochlea and then back out again, the exit point being the round window. Unlike a regular snail shell, which just has one coiled tube, the cochlea has multiple tubes inside of it. The two big tubes, the vestibular and tympanic ducts, are separated by a thin membrane of epithelial cells called the basilar membrane. This separates our cochlea into the two major chambers. If we slice through this snail shell and look at it head on, that's what this picture here is trying to represent. We have our two big chambers, the vestibular duct over here and the tympanic duct down here. This is the basilar membrane here, and there's even a third chamber in between the two big ones called the scala media, or the cochlear duct. It's in the basilar membrane where the hair cells are located that can convert the sound waves into action potentials. These are the receptors, or the cells that detect the sensation of sound. When we looked at the cochlea in cross-section, we noticed all of the hair cells were clustered in a region called the organ of Corti. But if you look at the entire cochlea, you notice this organ forms a spiral shape. We find hair cells along the entire length of the cochlea. The vibrations in the fluid entering the oval window will cause a structure called the tectorial membrane to vibrate, and when this pushes on the hair cells, that will open up ion channels, triggering an action potential. It's very important to take care of these hair cells in the organ of Corti because they do not regenerate. They are epithelial cells, so you might think they should be able to regenerate. But these are highly differentiated cells, meaning they've exited the cell cycle, and there are no stem cells in the organ of Corti to replace them if they get damaged. Like neurons, Excessive stimulation of these hair cells can lead to excitotoxicity. Too much calcium enters the cell, either triggering apoptosis or interacting with phosphate, causing crystals to precipitate, killing the cell. And when you lose hair cells, that leads to permanent hearing loss. There has been some research recently in trying to generate new stem cells to replace these damaged hair cells. And if you want to know more, go to this YouTube link down here and look into that research. Next, I need to discuss a little bit about sound so that we can understand how the hair cells convert sound waves into electrical signals. First, let's talk about a sound wave. If the air molecules in a room with no sound are evenly distributed, if a sound is happening, that sound is pushing some of the air molecules closer together so that they are higher density, followed by a space of lower density. Shown here is a sine wave, which is the simplest type of sound possible. And it would sound something like ooh. That would be a simple sound wave. We need to convert that into action potentials. One of the fundamental components of sound is its frequency. That's how rapidly the waves are hitting you. A low frequency sound would have waveforms far apart from one another, and that would be low in pitch. Whereas a high frequency sound would have waveforms very close to each other. Sound also has a volume or amplitude. Loud sounds, we say, have high amplitude. Quieter sounds, we say, would have lower amplitude. In addition to those two components, we're also going to have to worry about the phase of a sound. Imagine sitting in a swimming pool somewhere and two people jump in at opposite ends, creating waves in the water that travel towards you. If both of those waves hit you at the exact same time, you would actually experience a wave that's twice the size of either one. We would say that those two waves were in sync or in phase when they reached you. But imagine the two people jumped into the pool at slightly different times. So when the peak of one wave was hitting you, the trough of the other person's wave hit you. Those two waves could cancel out, and you wouldn't experience any change in the height of the water from where you were positioned. Those two waves would be out of phase, 180 degrees out of phase, completely canceling one another out. So we're going to have to worry about the waves hitting the left ear and the right ear, and whether they are in phase, out of phase, or somewhere in between. Keep in mind that that is three pieces of information. We have frequency, we have amplitude, and we have phase. And so far, I've only told you that the neurons leaving cranial nerve 8 can change the frequency of action potentials. One thing that's going to be weird is that the frequency of the sound is not going to be converted into frequency of action potentials. So things are going to get a little bit more complicated here in a moment. But it's going to get even more complicated. The sounds that I'm making now are multiple sine waves occurring all at the same time. Otherwise, my voice would sound something like ooh, ooh, ooh. And instead, I'm generating words. So to try and illustrate how complex sounds are really nothing more than sine waves, I've got a little video here that is going to convert some human speech and have it played back on a piano. Pianos don't generate perfect sine waves. There's a bit of a percussive or noisy sound right at the beginning, so it's not going to sound exactly like human speech. But pianos don't talk unless you play them properly, and that's what we're going to see here.
Let's see if the piano can reproduce a bit of Kennedy's famous inaugural speech. If that was a little hard to understand, let's try it again with the words on the screen.
Okay, so hopefully that illustrated that complex sounds like speech or music are really nothing more than a whole bunch of sine waves happening at the same time. Sine waves that have different frequencies, amplitudes, and may or may not be in or out of phase with one another. I bring that up because it's going to be an important factor in how the organ of Corti works. Remember, those hair cells were located along the entire basilar membrane that spirals around and around in that cochlea. This is where you find the hair cells. Let's unravel the cochlea. So now that it's just a length of tube, it turns out that this length of tube can absorb different frequencies of sounds at different places. High frequency sounds tend to hit the basilar membrane at the proximal end of the cochlea, whereas lower frequency sounds are harder to absorb. They travel further down the cochlea to hit the basilar membrane towards the distal end. You've probably experienced the same phenomena when you've listened to car stereos traveling down the street. It's very easy for you to hear the bass from a long distance away. You only hear the drums. And it's not until the car is really close that maybe you hear the rest of the music. That's because the high frequency sounds were getting absorbed by the environment very quickly. But the low frequency sounds were able to travel further. That's why you only heard the drums when the car was a block or two away. So the cochlea acts like a prism, only for sound. A prism can break up complex light waves into their individual sine waves. The cochlea can do the same thing. Let's say you're sitting in that swimming pool on your flotation device, and a skinny person and a very wide person jump into the pool at the same time. You're going to be bouncing up and down in a complicated fashion, both slowly and quickly. The cochlea can break up that complicated waveform into its individual components. When that complex wave hits the cochlea, the high-frequency component will hit the basilar membrane more proximally, whereas the lower frequency waveform will hit more distally. Action potentials will then be sent to the brain from these two locations along the basilar membrane, and it's the location now that is mapped to the brain in terms of frequency. The volume, or the amplitude of this waveform, would be represented by the frequency of action potentials traveling down these axons. So that's how two components of sound are translated into electrical activity. The frequency is represented by the location along the basilar membrane, and the amplitude is converted into frequency of action potentials. So watch out there, we have frequency used twice in that translation. Before we move on, I just want to point out that the hair cells are very specialized epithelial cells there that help to convert sound waves into electrical signals that can be transmitted to the brain along neurons. That is very different from ear hair. This man here holds the Guinness Book of World Records for the greatest amount of ear hair. We would not think that he has the world's best hearing because of his ear hair. So please be careful when discussing hair cells versus ear hair. We still haven't discussed phase. Right now we've only been talking about one ear or the other ear. But we receive the same sounds in both ears. If something is happening to the left side of our body, it will reach not only the left ear, but also the right ear. Our brain can compare the information from both ears to get some useful information out of it. For instance, the sound to our left reaches the left ear a little bit sooner than it reaches the right ear. So there's going to be a little bit of a gap. And the brain can compare the information coming from both ears to determine whether that is in phase or out of phase. And based off of the phase, it will perceive that as localization. Something from right in front of us would hit both ears at the same time. So when the sounds in both ears are in phase, the brain perceives that as coming from directly ahead of us. But when the phase is off, our brain can determine which ear the sound hit first and perceive that as localization. Once again, we've got an example of the difference between sensation versus perception. Our senses are that there's a millisecond delay in the sound reaching one ear versus the other ear. That's way too short of a time for us to be consciously aware of that. But our brain does perceive this information as location. It doesn't literally sound like a slight echo or delay from the left ear to the right ear. It sounds like something's coming from our left, or something is happening to our right. And this is known as spatial localization. And once again, it was the phase that was really important here. Are the waves happening at the same time, or are they slightly out of phase? And we're not aware of the phase, we perceive that as location. More goes into localization, though, than just the phase. For instance, we can tell when something is happening far away versus right up close. I could be whispering to you right up close, and even if I was quiet, you would know that I was really close to you. So the distance that a sound is coming from isn't just about its volume, but it's also about these things here, these tiny little echoes. If something is making a sound from way far away from us, it's also going to be bouncing off of a lot of other objects, and all of those bounces will be reaching our ears at a slightly different time. So all of these teeny tiny echoes tend to blur together in what we call reverb. If this red line represents the original sound, the sound tends to have a little bit of a tail. You can check it out right now. If you're in a small room, try clapping your hands and listening for the tail of the hand clap. The clap itself occurred in just a few milliseconds of time, but the sound tends to tail off afterwards. So try giving that a shot. See what the tail ends of sounds sound like in different rooms by clapping in them and listening to the reverberations. That is you sensing the reverberations, if you're actually listening to them. But we typically perceive this reverb not as a tail end of a sound, but it tells us a little bit about how close that sound is to our face or not. If the same sound was being produced right in our face, we would mostly just be hearing the sound itself, and it wouldn't have so many echoes, or they would be a lot quieter than the original sound. Things that are far away, though, the echoes sound about as loud as the original sound, and therefore we perceive that as appearing to be distant from us. The auricles also serve very important functions when it comes to sound localization. Sounds coming at us from the right side will reach the right ear unmolested. That sound wave doesn't have to travel through any skin, cartilage, or bone. But that sound wave, by the time it reaches our left ear, has traveled through some cartilage and bone. And that tends to filter out some of the higher frequencies. Just like the car stereos, the car door was able to filter out a bunch of the high frequency, but the low frequencies still reached you far away. Now that's what our ears are sensing, but we do not perceive that difference in frequencies, at least not literally. The way that our brain perceives this information is in location. Our auricles are very important for helping us to locate front and back. A sound wave coming directly in front of us or directly behind us would reach both ears at the same time. So phase is not going to help us here. This is where the auricles are very important. Sounds coming from behind us will tend to be filtered by the auricles before that sound wave reaches the tympanic membrane. And this helps us to locate things that are in front of us versus behind us. Without those auricles, we would have a much harder time locating sounds. High-frequency hearing loss is a big problem for many elderly patients. That's because high-frequency sounds have more waveforms per second and can therefore carry more energy. This can be problematic because it leads to excitotoxicity of hair cells. Therefore, the hair cells at the proximal end of the cochlea are more likely to get damaged than those at the distal end, leading to high-frequency hearing loss. This can not only cause problems with your patients being able to locate sounds, but are in their ability to understand human speech as well. So I've got an example of what removing high frequencies does to your ability to understand speech. The computer will be filtering out some of the high frequencies, mimicking high frequency hearing loss. And listen to what that does to your ability to understand what is being said.
1, 2, 3, 4, 5, 6, 7. Aha! You're on my apartment building on Granite Avenue. You owe me $300. Get it up?
Fred, take it easy. It's only a game.
Wilma, I'm just like them big tycoons. I play to win. Now, Barney, pay up or get out of the game.
That's 1 down and 2 to go. Betty, it's your turn.
I don't have any more money either. You got it all.
Then I'll take a mortgage on your orphan's home. Well, come on, take the dice, will you? Don't just sit there like a dummy.
And at this point, obviously, you can't understand anything that's being said. Most of the sound is still reaching your ears. There's a lot of low-frequency information reaching your ears just fine, but that's not enough to understand human speech. Our ears are not equally sensitive to different frequencies. It may have sounded like almost all of the sound was gone, when in reality, just most of the high-end information was lost. And it turns out our ears are most sensitive in the frequencies that the human mouth can generate. What this graph shows here is that the sound of my voice, it may only be 10 decibels in volume. To get the sound of a bass drum to sound just as loud, it would have to be 60 or 70 decibels in volume. So that's significantly louder. For that reason, the subwoofer on your stereo may be one of the largest components of your speaker system. It needs to generate a lot more force just to get those bass frequencies to sound as loud as some of the mid and high frequencies. To further illustrate just how complicated our senses are, damage to hair cells can not only cause hearing loss, it can cause people to hear sounds that don't exist. This symptom is called tinnitus and is often described as a persistent ringing in the ears. This can go from mildly annoying to completely debilitating and painful. Back when I was in school, this mechanism was completely misunderstood, and I was taught 180 degrees the wrong thing to try and explain how tinnitus worked. We now understand it better. Not great, but a lot better than when I was in school. Tinnitus can often be caused by overstimulation, loud sounds or noise. This can overstimulate the hair cells, which can cause the neurons connected to them to send really strong signals to the brain. And the brain can protect itself from excitotoxicity through adaptation. It can remove the number of receptors in the synapse over here in one of the many ganglia in our brain, down-regulating its sensitivity. Of course, if you do so much damage to the hair cells that they die, they then stop sending signals to the neurons at all. And now this synapse is way too inactive, and it leads to the opposite response. So after a few days of damage to the hair cells, this neuron in the brain may upregulate its receptors, making it even more sensitive, to the point where it starts firing action potentials, even though this neuron connected to its dendrites is not sending any signals at all. And now we hear a sound that does not exist. This is tinnitus. The solution for this? Well, unfortunately, these hair cells don't regenerate. But if those hair cells were just damaged, then the solution is to prevent them from being overstimulated and give them a chance to heal. get this person away from loud noises for a long enough period of time where the damaged hair cell could grow back, repair its damaged stereocilia, and ultimately replace this negative feedback loop here with the normal baseline situation. Another solution is to inject lidocaine directly into the brain, and that would just stop any action potentials from being created here in this ganglia. Next up, cochlear implants are a bionic device, tiny little microphones that can be placed on the side of the head that can convert sound waves into electrical signals, and then that electricity can be jammed right into cranial nerve 8. This can help restore hearing to anybody who has hearing loss in this area here, in the inner ear or maybe middle ear. It cannot help people with deafness that arises in the central nervous system. Now, our ability to convert sound waves into electrical signals is not exactly the same way that the cochlea does it. So this would sound very different to you or I, assuming that we all have hearing. But the signal can be sent and people can learn to understand it. For this reason, cochlear implants do best when implanted into young people whose brains are still plastic and have the highest capability of learning new things. It's much less successful to give a cochlear implant to an adult. Secondly, our ability to produce microphones to generate electrical signals is not nearly as complicated as the cochlea, and so those electrical signals are actually a lot simpler than a healthy human ear. My metaphor for this would be to glue a whole bunch of piano keys together. Remember, our cochlea, I said, was kind of like a piano roll, with the low notes on one side of it and the high notes detected elsewhere. A cochlear implant doesn't have nearly as many individual notes. We've glued a whole bunch of those together, trying to concentrate most of our effort right around the area where human speech is produced. But the ability to send low-frequency and high-frequency signals is a bit diminished. I can't tell you for sure what a cochlear implant sounds like, but based off of the technology, we have a pretty good idea of what it would sound like to somebody who has one. And on the next slide, I've got an example of that. It will go through a number of different channels, meaning how many piano notes do we actually have attached to our microphone. And it'll start very low, but as we get better and better with electronics technology, the number of channels in the cochlear implants is going up and up, which hopefully will be illustrated to you just how much that improves the ability to understand speech or appreciate music. So here we are. That has never existed. And same with that. We've never had as low as 4.
What kind of bait do you use to catch salmon? What kind of bait do you use to catch salmon? What kind of bait do you use to catch salmon?
What kind of bait do you use to catch salmon?
All right, that was speech. Try to guess what type of music this is. These are the older cochlear implants here. I could have sworn it was going to be a marching band, but that should help illustrate the number of channels and why having more channels leads to a better ability to understand speech and appreciate music. So that wraps up hearing and equilibrium and all of the parts of the inner ear. This would be a good spot to take a break, grab a snack, and come back for vision when you're done and ready. All right, on to the third and last part of this chapter, where we will discuss vision. In this section, we'll have to discuss trichromacy and why humans have a third cone, whereas most animals only have two types. And those that do have extra cones can see beyond what we call the visible spectrum of radiation, into the infrared or ultraviolet. Our third cone is pretty much redundant to one that other animals have. We'll talk about edge detection, and why humans are better at detecting the edges of objects, and why we might need that skill. We'll talk about peripheral vision being mostly in black and white. Even though it doesn't look black and white, most of our peripheral vision is actually detected by rods, which do not detect color. So we'll need to ask, why is it that everything in my peripheral vision looks like it's in color? And lastly, we'll talk about binocular vision. Why do some of us, animals, have two eyes in the front of our heads, whereas others have two eyes each on the sides of their heads? And what is commonly said is that it's for binocular vision. However, this is not the most true statement that we can make about having both eyes on the front of our faces. To do all of that, we'll start with some basic eye anatomy, and then we'll talk about what the eyes do in response to close versus distant vision. We'll talk about the cells in the retina that are responsible for converting light waves into action potentials, and how those signals are transmitted to the brain. In doing so, we'll talk about the molecule rhodopsin, and we'll talk about how this information is processed in the eyes before it ever reaches the brain. And to do that, we'll be discussing these weird things called on-center and off-center ganglia. And lastly, we'll also talk about the function of the optic chiasm. All of my other nerves are lateral pairs of nerves, whether they are cranial or spinal nerves. But the optic nerves come together to form an X, so we need to ask, why do they do that? Let's start with some accessory structures. There are a number of important glands that help to keep the eye moist. These include some tarsal glands found in the eyelids that help to prevent the eyelids from sticking to the eyes. There are a pair of pinkish blobs on the medial edges of the eyes called the lacrimal caruncles. These produce oily secretions which help to keep the eyes moist. Just like a good skin moisturizer, to help retain water we must have a hydrophobic barrier on the surface. So that's what these glands do, is produce those oily secretions that help to keep moisture in the eye. When these oils are not removed regularly by blinking of the eyes, they can collect into structures that anatomists like to call eye boogers that you might wake up with in the morning. Thirdly, the conjunctiva is a very thin epithelium that coats the surface of the cornea and the insides of the eyelids. This helps to form a barrier between the outside air and the eye itself. Of course, when infections are trapped just underneath this membrane, this can cause inflammation, known as conjunctivitis. The lacrimal glands produce tears. These glands are located on the lateral and superior sides of both of the eyes, and produce more watery secretions, which can help clean the surface of the eyes by washing away anything that gets trapped there. The secretions of the lacrimal glands contain antibacterial enzymes such as lysozyme. This is a digestive enzyme capable of breaking down carbohydrates. Of course, we are not digesting food in our eyes, but breaking down the cell walls of bacteria and viruses. When the lacrimal glands cannot produce enough tears, this can lead to irritation of the eyes. Artificial tears can help alleviate this. Artificial tears include water plus other chemicals, which makes the secretions a little bit thicker, such as cellulose or other polymers. In addition, certain types of acupuncture have been shown to increase tear production from the lacrimal glands. The eye is divided into two major chambers, with the lens being the divider between the two. Posterior to the lens is the vitreous body, a large mass of a gelatinous substance that helps to hold the eye in roughly a round shape. Anterior to the lens is the aqueous humor. This is a more water-filled chamber between the lens and the cornea. This fluid circulates around the lens, providing nutrients to the anterior portion. The eye itself can be further subdivided into three tunics, or layers: an outer fibrous tunic, a middle vascular tunic, and the innermost neural tunic. Let's start with the outermost, or the fibrous tunic. This includes the sclera, which is the white portion of our eyes. For most animals, you cannot see much of the sclera, if any at all, but for humans and a few other primates with no hair on their face, we actually can see a significant portion of the sclera. This allows us to engage in some nonverbal visual communication. I can tell where people are looking thanks to the fact it's easy to distinguish their sclera from the colored portions of their eye, the iris and pupil. The cornea, on the other hand, is also part of the fibrous tunic, but it is see-through. It's a non-keratinized, stratified squamous epithelium with plenty of collagen and other proteins, but it allows light to pass through, travel through the lens, through the vitreous humor, and hopefully hit the retina somewhere to be detected. We often don't think much about the corneas because they're see-through. Of course, when the cornea loses its transparency, then we think about it a lot. When the cornea gets damaged, this can lead to the formation of scar tissue, which is not clear. Also, vitamin A deficiency can lead to opaqueness developing within the cornea, which is the most common form of blindness worldwide, affecting over 10 million people. The prevention is medically very simple. Make sure people have enough vitamin A in their diet. But politically and mechanistically, this is not an easy task to accomplish. For people whose corneas have become damaged and have lost their clearness, artificial corneas or corneal transplants are an option. There aren't enough corneas donated year-round to give to everybody who needs them, which is why there's interest in the generation of artificial corneas, essentially see-through plastic that could replace the cornea without inducing an immune response. The corneas can be reshaped with laser surgery. This is known as LASIK. Changing the shape of the cornea can change how light is refracted as it passes through, the same way that light can be refracted as it passes through eyeglasses. And this can reduce or even eliminate the need for corrective lenses. Next is the vascular tunic, which contains three major portions: the choroid, shown here, the ciliary body, and most anteriorly is the iris. The choroid is the main portion of the vascular tunic. It's a layer of connective tissue which contains a lot of blood vessels, which provide nutrients to the other layers of the eye. For those that remember seeing red pupils in the days of flash photography, this is what you are actually seeing. The insides of the eyes are technically red. The pupils look black because very little light enters the eye to bounce back outwards to show us the red color. Next up is the iris, which contains smooth muscle cells. Some of these cells are organized in a circular orientation, the others are more longitudinal, and these can open and close the pupils. In reality, the pupils are just a gap. What is actually opening and closing is the iris itself. Here are some close-up photos of irises, showing the organization of collagen fibers. Even though we can't see the smooth muscle cell orientation, we can clearly see there are two sections to these irises, and these correlate to the circular and longitudinal bands of smooth muscle cells. Contraction of these muscle cells will lead to constriction or dilation of the pupils. We normally constrict the pupils to reduce the amount of light entering the eyes when we are in a bright light situation. On the other hand, when it's dark out, we will dilate the pupils to allow more light in, making it easier to see. Damage to the reflex that controls pupil diameter can be caused by damage to the brain or temporarily by drugs such as depressants. The ciliary body is a circle of smooth muscle traveling all the way around the lens. Constriction of this muscle will change the shape of the lens, which will alter how light is bent to hit the retina. It's a little weird how the ciliary muscles work. It turns out that when they contract, this relaxes pressure on the lens. Imagine we've got a bodybuilder here, and we've attached a bunch of bungee cords to his bicep muscles and stretched them up to the ceiling. When this bodybuilder flexed his biceps, causing them to bulge in the center, that would actually relax pressure on the bungee cords. And the ciliary muscles do the same thing. When they contract, it actually relaxes pressure on the lens and it becomes fatter. The lens itself is made of cells, but cells that have completely filled up with crystalline proteins. This means there's no room left for nucleuses or other stuff. Therefore, damage to the lens is very difficult to repair since most of the cells here are dead. A cataract is what happens when there's damage to the lens and it loses its transparency. The innermost portion is the neural tunic, which includes the retina and an outer pigmented layer. Let's start with the pigmented layer. This is a thin layer of cells that produce melanin. The function of melanin in our skin, of course, was to absorb UV light. The function of melanin in the midbrain was something that I didn't really cover, but it was there too. And here in the retina, melanin takes on a slightly different task. It's not protecting us from UV exposure and skin cancer, although it can do that. It's preventing light from scattering. If light were to enter through the eye and hit the retina, but not be absorbed by a rod or a cone, it might bounce off of whatever was behind it and hit other portions of the retina. And we would see that object as if it was coming from multiple locations. That's what having this pigmented epithelium does. If a wave of light is not absorbed by a rod or a cone, it will probably be absorbed by the pigmented epithelium behind it. One instance where we don't have enough melanin to absorb light is when we are driving at night and somebody's headlights come and blind us temporarily. Our pupils are very dilated because it's dark out, and suddenly these bright headlights shine a lot of light into our eyes, so much that some of it does not get absorbed by the melanin and it bounces around to other locations in the retina. And what should have been just two circles of light in our field of view now looks as if there are headlights everywhere, and you can only see white light. For people with albinism who can't produce enough melanin, that's a problem not just when they are driving at night, but all of the time. It can lead to difficulties in vision. Melanin serves other important functions, though, and people with albinism can have other visual problems, ones that couldn't be fixed by simply injecting melanin where it should be back there in the pigmented layer. And that's because melanin serves important roles in axon guidance. it helps instruct axons where to grow and connect. That's probably what it was doing there in the midbrain when we learned about substantia nigra and Parkinson's disease. The rest of the neural tunic is the retina, which includes 3 major layers of cells, plus a few other random cells. The rods and cones are at the back. These are the cells that will actually convert light signals into action potentials. Those signals would then be sent to the bipolar cells, there in the middle, and those would send the information to the ganglion cells. The axons of the ganglion cells ultimately become the optic nerves. There are some other important cell types in the retina, such as the amacrine and horizontal cells, but they weren't important enough to make it into my lecture. So if you read about them elsewhere, well, good for you. There's a region on the back of the retina that has no rod or cones. This is called the optic disc. All of the axons of the retinal ganglion cells have to leave the eye somewhere, and it turns out they all do that at the same spot, the optic disc. So this region of the retina, because it contains no rods or cones, if any light hits here, we cannot see it. So there's a little gap in our field of view for both eyes, all of the time. Why don't we see that gap? Well, you can trick your brain into seeing it, but otherwise the brain fills in that spot with information that it thinks should be there, such as whatever was there a half a second ago. One way to trick your blind spot is to take one of these strips here, focusing on the X, move this closer to your face until this red circle, the light from that red circle, is entirely hitting the blind spot. And at that point, your brain has to fill in that gap with what it thinks should be there. And it'll see that there's a green line to the left of it, hitting somewhere over here. And it'll see there's a green line to the right of it, hitting somewhere over here. And it'll just assume that that green line is the same green line. So at some point, this red spot will disappear if you're moving this closer to your face. And instead, it'll appear as one solid green line. If you've never visualized your blind spot, that's something we can do in lab, or you can do it at home. It's probably not going to work on the computer. You need to print this out or just make one with pens and paper. So if the blind spot represents where we have no rods or cones, the macula lutea is a portion of the retina that's very important to vision. It's within here that we've got a structure called the fovea centralis. The fovea is a microscopic structure, whereas the macula lutea you could see by shining a light on the back of somebody's retina and just looking with your own eyes. Either way, this is where we focus our vision, is on a tiny spot back in here called the fovea centralis. If this portion of the retina becomes damaged, this can cause a huge problem with vision. You're not going to be able to read any more than you would be able to read using your peripheral vision. That's because we focus everything right in this area. The fovea centralis contains a high number of cones. Those are for color vision. It also has very few of the other cells. Those cells got pushed to the sides here. In all of the other portions of the retina, light has to travel through the axons and the ganglion cells first, then through the bipolar cells, before finally reaching a rod or a cone down here. Now, in this picture here, we've stained the cells purple. In reality, they're fairly see-through, so light can do this just fine, but it still causes a little bit of fuzziness. In the fovea, however, there's not much to get in the way of light before it reaches these rods or cones here. This leads for higher acuity vision in this region of our vision, whereas everything else in the borders is more fuzzy. Alright, so that's some basic eye anatomy. including the chambers of the eye and the three major tunics, or layers, of the eye. Next, we're going to talk about some of their functions. Let's start with changes that occur in the eye when we switch from distance to near vision. When looking at distant objects, we need the lens to be flat and not bend light very much. To do this, the ciliary muscles would be relaxed. Therefore, the suspensory ligaments would be stretched tight, pulling the lens flat. Distant objects would then be in focus on the back of the retina. When objects move closer to us, we need to change the shape of the lens. We need to bend the light more. In order to do that, the ciliary muscles flex. That releases tension on the suspensory ligaments, and the lens adopts more of an oval shape, and this causes the light to bend differently, keeping the object in focus on the retina. Because this object is now closer to us, we can also constrict the pupils, reducing the amount of extraneous light entering the eye. What's more is that when objects move closer to us, both of our eyes will tend to converge on that object. That is accomplished by the contraction of the medial rectus muscles. So those are three processes that occur in the eye when we switch from distance to near vision. Let's move on and talk about the cell biology of how we convert light waves into action potentials. That is accomplished by two cell types, the cones, of which we have three types for color vision, and the rods, which are larger and are used more for black and white vision. Both rods and cones have an outer segment full of membranous discs, and then an inner segment which has all of our basic cell stuff, like the nucleus and mitochondria. It's those membranous discs where all of the magic occurs. The membranous discs are composed of phospholipid bilayers, in which are a number of G-protein coupled receptors called opsin. To help convert light waves into a second messenger system, retinol, a molecule synthesized from vitamin A, is also a component, and together these make up the molecule rhodopsin. The retinol molecule can switch from a cis to trans form when it absorbs light. And when this happens, it'll induce a conformational change in the protein, which bumps off an activated G protein. And then we get our standard activation of a second messenger system. And as we remember from earlier in the term, second messenger systems are good at amplifying signals. This is helpful for us to be able to detect low levels of light. For those that would like to learn a little bit more about genetic engineering, we can use rhodopsin to confer photosensitivity to other cells. For instance, if people have a form of blindness caused by damage to rods and cones, inducing the expression of rhodopsin in some of the other cells in the retina, like the ganglion cells, can convert them into light-detecting cells, helping to restore some vision to those people. A photon of light can induce a conformational change in retinol, and this activated rhodopsin. But when the light goes away, the retinol does not switch back to its original conformation. To get this rhodopsin ready to absorb another photon of light, we must kick out the old retinol and replace it with a new one. Luckily, rods and cones have plenty of retinol, and they should never run out under normal situations. But we can induce an abnormal situation. For instance, if you stare at this image over here to the right for a minute or two without moving your eyes much, you will overstimulate some of the cones in your retina. For instance, the blue lines will be bleaching out the blue cones, but not the red or the green ones, in certain regions of the retina. And when you look away and fix your gaze on a white piece of paper, instead of looking white, because all three of your cones are stimulated, the blue ones have been bleached in certain areas, and that will make those areas of your retina appear red. I've got a diagram over here that should hopefully illustrate that a little bit better. As we stare at the object, the American flag that had blue lines and the yellow border around the stars. This is going to photobleach certain cones in certain areas of the retina, which I've illustrated simply here on the right-hand side. So when blue light hits certain part of the retinas, it'll be activating the blue cones, and yellow light hitting other parts of the retina will be activating the red and the green cones. And it's going to be those cones just in those areas that get bleached after a few minutes of staring at this object. In other regions of the retina, for instance these black bars, the cones are not being overstimulated at all. So if we bleach out just a few of the cones in a few places, and then look at a white object, white light should stimulate all of the different cones, but some of our cones are not going to respond. So in the area up here, if just the red and the green cones are activated, the blue ones being bleached out, the brain will interpret this now as a red stripe. And down here, where only the blue cones are activated because of the red and the green ones have been bleached out, the brain will interpret this as a blue border around the stars. Hence, we now see the American flag. The rods and cones don't have to make the same amount of rhodopsin all the time. They can upregulate its expression and downregulate its expression. And they do this in order to adapt to highlight or low light situations. We can sense 1 billion different shades of light, but only about 1,000 different ones at any given time. And we just shift that range of sensitivity to darker or brighter shades. Now, it takes longer to make rhodopsin than it takes to remove it and break it down. Therefore, it takes longer for us to become adapted to the dark than it does to become adapted to a very bright situation. For anybody who's ever watched Mythbusters, they did a very great illustration of why pirates might have worn eye patches for the ability to fight on the deck of a ship and then quickly move down below and not have to wait 30 minutes to become accustomed to the dark, because of course they would have been stabbed to death quickly. Still, we need to answer the question, why these two types of cones here, the green and the red, that seem to overlap? So what this image to the right here shows are the different wavelengths of light that can stimulate the 3 different types of cones. And what we see is that there is some overlap. A blue cone and a green cone and a red cone are named because those are the wavelengths of light that are best at stimulating these different cones. But even a 400 nanometer wavelength here would still slightly stimulate one of the blue cones, although not green or red. Conversely, something around 650 nanometers in wavelength would barely activate a green cone, activate a red cone a little bit better. But why this overlap, this extreme amount of overlap between the reds and the greens? And in fact, most mammals only have 2 types of cones. We've adapted this 3rd type, so what does it do for us? Now, some animals will have a cone over here that allows them to see into the ultraviolet spectrum, especially birds and insects that might pollinate flowers. Other animals might have a 3rd type of cone over here that allows them to see in the infrared, especially those that hunt at night. But we've got this 3rd cone that's smack dab in the middle. It doesn't allow us to see longer wavelength or shorter wavelength colors. It allows us to differentiate between some of these wavelengths with a higher degree of accuracy. So what does that mean? Well, let's take a look at this image here to the right. This is a representation of a spectrophotometer. And what it's telling us is that blue wavelengths are here, yellows and greens in the middle, and reds are over here. And these are the wavelengths that are reflected by human skin, either Caucasian or African American or Asian skin. And what this tells us is that we're all pretty much the same skin color. Some of us have a little bit more absorption than others, But otherwise, we all have the same three pigments. We've got kind of a reddish-bluish from our blood, a more yellowish and orangish from carotene, and more of a brownish from melanin, which will be a mixture of different wavelengths. Our third cone allows us to differentiate between slight differences in skin color a lot better. And so while this machine says that human skin is all pretty much the same color, we typically do not. In fact, we assign words for different skin colors that are the exact opposite of one another, like black and white. There could be no more extreme difference in colors than black versus white. And yet, in reality, our skin colors are all very similar. So do we have this third type of cone so that we can tell the difference between people of different races? No, that's not what it's for. It's for us to be able to detect things like blushing and other emotional changes that might affect skin tone. Or we might want to be interested in health changes to the skin. We are a society, after all. We might be interested in when our friends and family are sick, and we could tell that if their skin was a little bit cyanotic or a little bit jaundiced. If it's a stranger who's jaundiced, we might not want to let them into our community, and we might shun them. And why do we care about these emotions? Well, unlike most other mammals, human females do not, let me see how to say this, advertise when they are ovulating. So we are known as cryptic ovulators. So to figure out when men and women might be interested in breeding, we need to rely on more emotional changes. We're not very good at detecting pheromones, our skin color doesn't change drastically, and we don't produce different scents at different times when we go through heat, the way that other animals do. So it's very important for our ability to reproduce to be able to detect slight changes in emotional status. You may have learned in a genetics class that missing one of the types of cones could lead to a defect in vision known as color blindness. These are usually genetically recessive traits, often sex-linked, which makes for really fun Punnett squares. But is it really a defect? If it was truly a defect, we probably wouldn't see so many people with color blindness, no pun intended. So why might some people be colorblind? Well, for people who are colorblind, for instance, this person's fovea centralis has a lot more green cones because they have a mutation that prevents them from making their red cones. So now imagine what colors this person would be better at detecting. And the answer is they could detect finer shades of green and be able to distinguish between them, even though they can't detect red. And that is really beneficial when you might be trying to look and spot camouflage or animals that are trying to blend in with their environment if you're a hunter. Because humans have often hunted in packs, it would make sense to have one person in your group who is colorblind, but you don't need everybody in your group to be colorblind. And we see that roughly 10% of people, 10% of men have some form of color blindness. And so if we assume that hunting groups historically have been about 10 men in size, then on average there should roughly be one in every group that has color blindness, and therefore might be better at detecting certain types of animals as they try and blend in with the environment, and point them out to the rest of the group. In fact, in World War II, colorblind men were actively recruited by the Air Force and the Army for their increased ability to spot camouflage targets. You didn't need everybody in your unit or the bomber to be colorblind. It just took one to point out where the object was. So, in reality, colorblindness is probably more of an adaptation. And because we are a society, we see that not everybody is colorblind, but having some people colorblind in our groups has a certain benefit. Let's review some of the concepts that we've covered and focus on how this explains the difference in sensitivity between rods and cones. We've said that rods and cones both use a rhodopsin-type molecule, but rods are bigger than cones. Therefore, they've got more rhodopsin. What's more is that there are more rods than cones, and the rods will respond to any wavelength of light, whereas cones only respond to certain wavelengths of light. These are 3 reasons why rods are more sensitive to low levels of light than cones, meaning if it's dark out, you're probably not seeing very much in color. You're probably seeing mostly in black and white. The 4th reason why rods are more sensitive than cones is that they exhibit a lot more summation onto the ganglion cells, whereas the cones more or less synapse on the ganglion cells in a 1-to-1 format. It's this summation of the rods that we're going to talk about next, and can help explain why rods are better at detecting motion than cones, meaning you're going to be better at seeing things move in your peripheral vision than you are if you focus on the fovea centralis. The summation of rods onto the ganglion cells is complicated. Some of the bipolar cells will trigger EPSPs and others IPSPs. We're going to call these organizational patterns on-center ganglia and off-center ganglia. And let me focus on just the on-center ganglia, and the pattern of the other one will just be the exact opposite. For this on-center ganglion cell to be stimulated, we must activate only the rod in the center. This will lead to activation of the ganglion cell. We want the rods on the borders to remain unstimulated. That's because the bipolar cells that they are connected to would release inhibitory neurotransmitters onto the ganglion cell. Therefore, if we stimulated all of the rods, we would inhibit the firing of action potentials by the ganglion cell. So this ganglion cell will only fire action potentials when the center rod is stimulated, but not the border rods. That's why it's called an on-center ganglion. That doesn't answer the question of why these are wired up this way. And to illustrate that, let's go to the next slide. Imagine we are looking at the retina head-on, and these are all the tops of a bunch of rods, and we shine a circle of white light onto them. Many of those rods would be activated strongly, some would be activated weakly, and others not activated at all. If these were cones, this is the sort of information that would be sent to the brain, a literal representation. But rods are organized in those on-center and off-center ganglia. So let's look at just 2 of these rods, one on the border and one in the center, and what will be happening to the information that these send to the brain. The one in the center there is surrounded by other rods that are also activated, so they will be generating IPSPs onto the ganglion cell, and therefore information from the center is not sent to the brain, even though the rods were activated here. On the edges, though, the rod in the center is stimulated, but most of the border rods are not stimulated. Therefore, we're actually going to enhance the signal from this area. And the net result is that the ganglion cells in the center aren't going to be sending much information, but the ones on the edges will. We have detected the borders of this object. And that's one thing that rods, being hooked up to on-center and off-center ganglia, will allow us to be able to do, to highlight the borders of objects. That allows us to see shapes just from outlines. For instance, if you look at the image here to the left, I'm sure all of you see 6 dolphins. You don't? what do you see? Well, you've got a dirty mind. Here, let me highlight one of those dolphins for you. There, now can you see the dolphins? All right, so this is a bit of an optical illusion that also illustrates how what we see depends on our life experiences. Children, when they look at this, typically only see the dolphins. And yet now that we've got the outline of one of these objects, it should be pretty easy to spot these aquatic mammals. Rorschach blots are the same type of information. We see images in just blobs of ink. If you give us just the outline of an object, our brain is able to tell what that object is because we're used to dealing with just outlines. This helps us when we're hunting. For instance, my cat has great difficulty tracking grasshoppers when they land in the grass. He can't see them anymore. I can't though, and that's because my rods are outlining the object. They are telling me where the green grasshopper ends and the slightly different shade of green grass begins. The on-center and off-center ganglia will also explain this optical illusion here to the left. If you focus on any of the intersections, they'll appear white, but all of the others in your peripheral vision will appear to have a grayish circle in them. But if you move your eye around and look at different intersections, now they appear white, and the one you're no longer focusing on has a grayish circle. So why do the ones that we focus on always appear white, and the ones on the borders always appear to have a grayish circle? Well, that's because when we focus on one of the intersections, the light from that image is hitting the fovea centralis, which relays fairly literal information to the brain, and it's telling us exactly what is there, a white crosshair. But in my peripheral vision, all of the other intersections are mostly hitting rods, which are hooked up into on-center and off-center ganglia. And so in most of the other intersections, the rods in the center are stimulated, but so are a number of them on the borders. Not all of them, but many of them, and so that tends to inhibit the signal. Hence, it appears a little bit grayish at all of those other intersections. None of the other places appear gray, no matter where we're focused, and that's because those rods, stimulated here in the white areas, most of their neighbors are in a dark area, and therefore we are not inhibiting the signal from here. The majority of the retina is for peripheral vision, and it's mostly rods located in these areas, and they are hooked up to the ganglion cells in the on-center and off-center conformation, which allows for summation. Hence, our peripheral vision is mostly in black and white, but it's also more sensitive to dim light. If you are a stargazer and you want to see a fairly dim object in the night sky, don't try looking straight at it, but look just off to the side, and you'll be able to detect that dim light a little bit better in your peripheral vision. On the other hand, if you want to get color information, you need to focus on that object. And if you want high resolution, you need to focus on that object. We cannot read using our peripheral vision. For that, we must focus on just a few letters at a time using the fovea centralis, which is mostly cones, and they are wired up to the ganglion cells in a 1-to-1 conformation, which means they're less sensitive, but that higher resolution means we get less blurriness when we do see objects. Next, we need to discuss the optic chiasm. The two optic nerves, as they leave the eyes, join to form this X-shaped optic chiasm, and then two optic tracts extend back to the brain to connect to the thalamus, which will relay this information to the occipital lobe. None of the other cranial nerves do this. They just connect to one side of the brain or the other side of the brain. So why do the optic nerves connect at this chiasm? And why do the nerve fibers hemidecussate? That means half of them cross over and half do not. And that's to keep the brain wired in an organized fashion. It turns out that the left side of both retinas connect to the left side of the brain. That means the left side of the retina of the right eye must cross over to connect to the other side of the brain. But the left side of the left eye does not. It stays on the same side. This is hemidecussation. That means objects to my right will be detected by the left side of the brain, whereas objects to my left, as you may have guessed, will be detected by the right side of the brain. So unlike my arms and legs, most of my body was connected from the right side of the body to the left side of the brain, and the left side of the body to the right side of the brain. The eyes, their wiring is special. It's the left field of view that's connected to the right side of the brain, and the right field of view to the left side of the brain. Objects in the center will be detected by both sides of the brain. And rather than seeing two images, the brain will consolidate these two separate pieces of information into a single image. Usually one side of the brain is a little bit dominant. That's why if you close one of your eyes, you don't notice much of a change. But then when you close the other eye, it seems like your vision shifts a little bit. One of your eyes was the dominant one, and we're shifting the other eye to overlap what we see in the dominant eye. Coming up next, I've got a bit of an optical illusion where we can force the brain to try and fuse two images that should not be fused. Keep staring at that little cross there, and try and look at these famous actors and actresses' faces in your peripheral vision. And what your brain is trying to do is fuse them into a single image. And it's not doing so very well. I'll let this run for a few more seconds. Okay, let's move on. To consolidate images seen in the left and the right sides of the brain requires a functional corpus callosum. A few people might have their corpus callosum severed to help reduce the severity of epileptic seizures that they suffer from. And as I understand, that leads to very little visual deficits. In fact, the only time that they would notice that the left side and the right sides of their brain are not communicating properly is if you trick them. You have to show them an image up close. For instance, in an experiment done at Caltech, people with severed corpus callosums were shown images of pirates in which the pirate had an earring in just one ear. If that earring was over in this ear over here, it was detected on these sides of the retinas and therefore only sent to this side of the brain. Normally, this side of the brain would have told the other side what it saw. But now if the right side of the brain is the only side that actually gets the information about the earring, and you ask these people about the pirate that they see, they will tell you they see a pirate with no earrings. That's because language centers are more on the left side of the brain typically. But if you ask these individuals to draw what they will see, they will draw a picture of a pirate with an earring in their right ear. And even after they've done that, they will still not be able to explain to you in words why they drew the earring in the right ear of that pirate. Because this side, the left side of the brain, did not get that information. One more interesting feature of the on-center and off-center ganglia is that means that the retina sends information to the brain about relative shades, not absolute shades. For instance, if you look at the two center squares, they are actually both the same shade of gray, but they will appear different to you, at least until I cover up their borders, and now it becomes much more clear that they are the same shade. When on-center ganglia are activated and suddenly turn off, and an off-center ganglia is activated right there, we detect that as motion. We've switched from light to dark to light, as if a dark object has just moved over a bit, or maybe a light object has moved a bit. For instance, if you look at the circles, it may appear as if that pink circle was moving, when in reality it was just circles changing from light to dark and dark back to light. Our brain perceived that as motion. What motion really would look like, a pink circle moving around. Now there's literally a pink circle moving, not just circles flashing on and off in a pattern that mimics motion. Therefore, if you want to detect motion, it's better to use your peripheral vision. Do not stare directly at the object or in the direction that you think something is moving. Stare slightly off, and you will be better able to detect slight motions that way. Here's a couple more optical illusions, just for fun, that should help illustrate that our brain is more interested in relative shades than absolute. On the object to the left, squares A and B are absolutely the same shade of gray, but they look different to us. Let me cover over this image just to show you that those are the same shade. Now as we go back, they appear different once again. And even though you know they're the same shade, we can't change this. So what's happening here? Well, it's a little bit of on-center and off-center ganglia. The borders are a little bit darker or lighter, respectively. But another thing that's different about square B is that it appears to be in a shadow. And so our brain is subtracting out some of the darkness, saying, well, yes, it appears darker, but in reality, it should be lighter. It just looks darker because it's in a shadow. And so after your brain has subtracted out what it thinks is the darkness due to the shadow, square B now appears lighter. This is the same reason that people might have seen white stripes or other people blue stripes in that infamous dress that either had, what was it, black and white stripes or blue and gold stripes. For those that saw blue stripes, that was probably closer to what was literally there in the image. But for those of us that saw white stripes, that meant that our brains were saying that, hey, sure, that does look blue, but it's in a shadow, so that bluish probably comes from the shadow. And so we saw it more what our brain thought was the absolute or real color, which was white stripes. Once again, let's move that back over, just to show us that these two boxes really were the same shade. Over to the right, I have another fun optical illusion. Depending on whether you're focused on the little plus sign in the center, or whether you're looking at the circle that appears to be moving, it'll either be disappearing, or it'll be turning green. I'm not really sure why this one works. I just thought it was fun. Many optical illusions work because the information was altered in the retina before it was even sent to the brain. So when we see the dark circles where there shouldn't be dark circles, that wasn't a difference between sensation and perception. We really were getting that information from our retina. On the other hand, some optical illusions work because the brain perceives that information differently from how we sense it. For instance, the blind spot, or the optic disc, is an area of the retina that never sends light information to the brain, and the brain just fills in that gap all the time with what it assumes should be there. The next optical illusion that I'm going to show you will illustrate that most of our peripheral vision is in black and white. We do have a few cones scattered through the retina in our peripheral vision, and that can help the brain to fill in what is mostly black and white information with the colors that should be there. The next illusion is going to trick our brain into doing it when it shouldn't. Go ahead and focus on that black dot in the center of this image. It's okay to blink, but try not to move your eyes around too much. If we keep focusing on this black dot, eventually this photo is going to change and it should appear to be a color photo. When that change occurs, I want you to wait until you're sure it's a color photograph, then look away and look back and see what you see. I'm not going to tell you exactly when to look away, pick some time to look away and then look back. And hopefully you've all done that now. And what was appearing to be a color photograph a second ago is actually a black and white photograph. You can hit rewind and see that when the image changed, it changed to black and white. And our brain was tricked into filling in all of our peripheral vision with color that it thought should be there. And it did so. But when we looked away and looked back, and our fovea centralis alerted the brain that no, actually, even all of these edges here are in black and white, the brain stopped filling in that color information, and we lost the illusion. It now appears as just a black and white image, no matter how long you stare at it. Let's summarize. The most superficial part of the retina was the pigmented epithelium. We then have rods and cones, bipolar cells, and the retinal ganglion cells, whose axons travel over the surface of everything and then exit the retina to form cranial nerve 2 at a location called the optic disc, or our blind spot. When light passes through the cornea and through the lens, it should be focused on the rods or cones, which will be stimulated and activate the bipolar cells to activate the retinal ganglion cells, which will fire an action potential, which will travel to the thalamus and be relayed to the occipital lobe. The fovea centralis sent a very literal representation of what colors of light were being detected and where. In our peripheral vision, however, a lot of this information was changed before it reached the brain. The rods, because they summed onto the on-center and off-center ganglion cells, would only send information where the colors changed from light to dark, or from dark to light. When this information reached the brain, it was further altered before we came to a perception. For instance, the brain filled in our blind spot, it colored in a bunch of our peripheral vision, and it consolidated the images from the left and the right side into just a single image. That will lead us to our next question. Why do we have two eyes on the front of our head when many animals, like cows, have eyes on the sides of their head? That limits how much we can see. There must be a benefit. And most of us are told this is for binocular vision, or depth perception. However, if you close one eye, it's not as if the world suddenly seems two-dimensional. Having two eyes in the front of our head can help with depth perception, allowing us to triangulate how distant objects are away from us. But in fact, most of our depth perception does not come from having two eyes at all. For instance, if you look at these two images to the right, the only difference between the two is where the shadows are located, and that changes what we perceive greatly. The upper image appears to have 4 spheres at different distances, but the lower image appears to have 4 spheres at the same distance, but at different heights. So where shadows are located helps us to determine how distant objects are away from us. And you can close one eye or the other eye, and that will not change this illusion at all. It does not take 2 eyes to have this sort of depth perception. Another example of how our brain is more interested in relative information than absolute is illustrated here. These 3 images of people are absolutely the same height, but relatively, the one in the back appears to be much taller. That's because our brain compensates for what it thinks should be changes in size due to distance. People who are far away should appear smaller than they really are. And rather than our brain thinking that those people are small, our brain says, nope, it's a normal-sized person in the distance. Here, we see this person in the distance as appearing much larger than they really are. We think that because they're in the distance, they should appear shorter, and therefore our brain tries to compensate by making them look larger. This, once again, does not take 2 eyes. If you close one eye, this illusion of depth perception still works. Knowing those last 2 things, we can now explain a little bit about why this man's street art appears to have a portion of it floating in midair. A, he's painted the ground to appear to have shadows, and we're interpreting that as some sort of distance. B, his letters are changing in size along these walls as they travel further away from us, and our brain tries to compensate for that distance by perceiving the letters differently. And he's painted his letters to perfectly match this adaptation. And so now the letters all appear the same size and the same distance away. What's literally letters on walls receding away from us now appear as letters floating in air going across and between the two walls. So why do we have two eyes in the front of our head if we don't really need that for depth perception? And if it was absolutely necessary for depth perception, people who had lost vision in one eye would probably get in a lot more car crashes, and therefore would be charged higher insurance rates. Insurance companies are very good at risk assessment. They know when people are more likely to commit accidents or not. And people with only one eye are not charged a higher rate for car insurance. Young men are, but not one-eyed people. So, that should also help illustrate that it does not take two eyes to have very good depth perception. So why do we have two eyes? Well, if you have two eyes, put your fingers in front of them and stare at a distant object. You can still see it pretty well. And that's because your two retinas are getting slightly different information, because they are at slightly different angles from that different object. But the brain consolidates that information, and you can more or less see through your fingers. Close one eye, though, and that ability is lost. That distant object may now be hidden from you. We always like to play a fun game called Nobody Home and stick your thumb in front of your face so that you don't see the person and you can pretend that they're not there. Having two eyes open, that would not work. You could still see the person with your other eye, and your brain would essentially be seeing through your thumb to look at that distant object. And that would be very beneficial if you were hiding in some grass or amongst some branches, but you were trying to look at animals that you were hunting that were far away from you. If you only had one eye on one side of your head, the grass or the branches might obstruct your vision, and it would be more difficult to track that animal that you were hunting. So having two eyes on the front of our head allows us to see through the underbrush better and keep a focus on distant objects that we are interested in, even though close objects might be partially in the way. So that's going to wrap us up. We talked about the rods and the cones and how they were different, the amount of rhodopsin, whether it detected all wavelengths of light or just certain colors, how those cells summed onto the bipolar and ganglion cells. The cones were pretty much one-to-one, but the rods might be hooked up into an on-center or an off-center conformation. We talked about differences between sensation and perception. The brain did not perceive the blind spot because it filled in those gaps, for instance. And we talked about the optic chiasm, keeping our left field of view and right field of view sent to just one side of the brain or the other, rather than split half and half. Earlier, we talked about the accessory structures, the three basic layers or tunics of the eye, and the three major changes that occur when we switch from distance to close vision, namely convergence, constriction, and accommodation. But that's going to wrap us up for Chapter 14.